is Kevin Romer. I'm past president of National Tay-Sachs and a very, very proud father of Matthew Romer, who was diagnosed and was our reason for joining this organization back in 1996. He was diagnosed with infantile Tay-Sachs disease. And he was really quite an inspiration for us. In 1998, we established an organization in his honor called the Matthew Forbes Romer Foundation and became an affiliate of the National Tay-Sachs and Allied Diseases Organization for the state of Florida. And we worked hard on a lot of the same missions and delivering them locally and working nationally. And in around 2002, 2003, Matthew lost his battle. He was supposed to live till about 3 or 4 years old. He almost made it to his 8th birthday. He was an incredible fighter, an incredible inspiration, and quite frankly, the reason why around that same time we partnered with the national organization to create the National Research Initiative. And so we're very proud to be here to give you an overview of our journey together. And you can see that's little Matthew in the middle. It's a beautiful picture. His wife, my wife, his mommy. He was very precocious for his age. Lisa Jane sends her regrets and love to so many of you who she's met over the years. And Jeffrey and Alexis, who are not able to be here either, give their love too. So it's great to be here and as a part of this important session. You know, when we were talking about research, there's so much important science and so much important progress that's been made, but that's all irrelevant unless it all begins with the human side of it. And how does this relate to all of us here in this room? And you can see in the upper left where we have the beautiful Rachel Epstein with the Canavan disease, who's fighting her battle with Dr. Gao's lab at the UMass. And what's even more exciting is Sister Jessie, who's a part of the team. She's been inspired to join the fight, and she's working in the lab there too in the back. To the right, you can see Doctors Martin and Bradbury, who are part of an incredible team. And propped in front there, they're leading the charge, of course, is Porter Heatherly. And Porter unfortunately lost his battle this past year to GM1. And again, is a reminder of how short our window of opportunity is and how urgent our mission is and how many people on that team are inspired by children like him. In the lower left, we have Owen Vogel with juvenile Tay-Sachs disease, propped on the shoulder of one of our heroes. Everybody's in this room is a hero, but Cynthia Tift, who's been there from the beginning of this fight, she was our first scientific advisory chairperson, and she really helped to build this research initiative. And she has some very, very burning desires to see us reach our important goals. And in the lower right, we have our families from late onset that are working with the clinical assessments that are going on right now that are so important for some of these small molecule projects that we've been doing over the years. And so you're saying, well, we talked about the human side of research, right? So what's that thing in the middle there? Okay, so who's watching March Madness right now? Nobody? I didn't think. It's not that big a deal, right? Every team has a mascot, right? Correct? Our research initiative is today announcing that we have our new mascot. It's the sheep. Right? So we're gonna go forward with the sheep leading. But here's a little secret. Most people don't know this, that inside that's really a uniform, an outfit. And our president, Brian Manning, dresses up as the mascot. You don't see him now. He's actually, oh, yeah, okay. So next time you see the sheep, say, Brian, how's it going? So, but it really is all about the people, right? Okay. But it's also all about progress and results. And we're standing up here today, 15 years later, after starting the National Research Initiative, to talk about real results. And where are we going from here? Now, let's not forget the fact we're a 60-year-old organization. Our roots go back to some very special pioneering work that was done in the 60s, 70s, and 80s to understand the deficiencies of the enzymes to understand and identify what the gene itself was, to be able to work with it. But we set out 15 years ago to say, okay, we have to be on a trajectory that is a rocket ship, and we got to get there. And so we began in 2002. $3.5 million of grants have been funded by this team here, by the families, by the people that are so passionate about what we're doing. 54 projects. And Sue will talk about what this really means about the whole spectrum, the whole pipeline. If you think about how drugs and therapies are developed, we have really driven a complete spectrum of that. And what's great about it is we've been a catalyst for so much more, because with all the work that we've done, it has spawned follow-on work that's been funded by the NIH to over $20 million, all being directed towards the work that we're doing. So congratulations to all of you for your contribution to this and your belief in this mission that we're on. It's my pleasure now to introduce Sue Kahn, our Executive Director, to present the rest of the presentation.
Good morning. So what you see here is a general picture of the drug development process, and I wanted to use this to explain what types of grants and what types of research NTSAD funds. So I'm not going to go in detail about what the drug development process is, but this puts it in context. I mean, at the very beginning,
drug discovery, the first steps. What our role is we provide what's called seed grants, small grants, so that investigators, researchers can gather enough data to then go to the NIH. It could be a European funding organization to get a grant of a more significant size. And in this note, I'll put in a little plug for the advocacy session, because for those of you who read the headlines, there's a proposed cuts in the NIH budget, and that's not good for anybody. So we want to maintain that research. So just a little side note. Then further along, as we get into what's called the preclinical stage, where we're really developing the data that shows we have a potential drug, it's working in animals. The NIH, again, other larger sources of funding can be available, but we get grant proposals in these different areas that are listed. In animal models that are needed, because before you go into humans, you want to show that it works in animals. What's called preclinical efficacy studies, safety and toxicology studies. These are all, again, types of projects that maybe there are larger grants available, but all these, to be able to apply for these larger grants, you need data, you need experiments, you need to be able to apply for those grants. And that's really where NTSAD and other foundations come in, where we have the ability to provide these unique gaps. We look at what are kinds of projects that other groups would not fund, where can we uniquely help in, whether it's Tay-Sachs, Sandhoff, GM1, Canavan disease, that if we don't do it, we think it can advance. We want to be fill that need. Other types of projects that we fund, biomarkers, which are used to measure how the disease progresses, if a treatment could be working, what we call natural history studies, and then also newborn screening. So those are just some of the types of projects that we fund. And just to then link that to what we're going to talk about today, the first panel, which is going to be moderated by our Scientific Advisory Committee Chair, Fran Platt, is going to be about gene therapy, and that gives you, hopefully you'll get to understand what is the state of gene therapy today, where is it going. You'll have a chance to ask questions as well. We have 4 distinguished panelists. And then we have what we're calling, as we move kind of further along the drug development arrow, we have what's called the clinical trial readiness panel. What's clinical trial readiness? Why is it important? How can we fill the gap? where there's knowledge that's necessary to be gained to work towards clinical trials. And that panel will be led by Dr. Cynthia Tift, who was sitting right there. Oh, okay. But I do want to put in a plug because this year NTSCD nominated Cindy for what's called the Rare Impact Award for the National Organization of Rare Diseases. This is a very big deal. It's very competitive. She was picked to receive this award in front of 1,000 people on May 18th. And we want you, many of you know her, you've worked with her, we want you to be part of the celebration. So Jean's gonna arrange, so we want some photographs for you to be taken with Cindy, so that on May 18th we'll see you at the celebration. So again, not to divert this conversation too much, in Washington, D.C. So if you're interested in attending, I'll send out information. Maybe I can get you a seat somewhere. And then lastly, to complete this, we're going to have Corliss Schwartz from Sanofi Genzyme speak about the regulatory process. What's it all about? How do drugs get approved? I know that we've received a lot of questions about this process. So hopefully you'll have an opportunity to hear answers to some of the questions you've been thinking about, and then also ask your own questions. So here's the agenda, but two things I want to point out. In past years, we had the scientists present information, and this year we decided not to have our typical scientific presentations. But what we did do is we prepared this book, which all of you should have received, where we summarize not only some of the grants that NTSCD is funding, but also grants that other foundations are funding. And research and making progress is a team sport. So none of us are going to do it alone. We have to work together. We're all working towards the same goals. So on the front of this booklet, you'll see some of the logos of the foundations who also work to advance research in our diseases. And the agenda is in your booklet, but I think at this point, I will turn it over to Fran, who's going to give an overview of therapeutic approaches to our diseases.
Thank you. Well, it's a great pleasure to be here. I have jet lag, so bear with me. I got in last night, and as you may hear from my voice, I've come from the UK. So my job is to really just introduce to you some of the approaches that can be taken to treat this family of diseases, and give you a snapshot of where the progress is, and also to alert you to a few emerging things that may be very relevant to this community in the coming years. So my title is What, Why and How, and I'm going to start with the what, and that is what's the basis for the diseases that we're here to discuss. So as you're all aware that the Tay-Sachs and related diseases are genetic diseases, and you may hear about, if you read any newspaper, you may hear about other forms of genetic diseases that are out there, and they really fall into two categories: complex genetic diseases. This is where you don't have a single gene that causes the disease, but where you may have a gene where, when it's inherited, gives you a greater risk factor. So, for example, you may have a greater risk factor of diabetes or heart disease or Alzheimer's, but by having that gene mutation, it doesn't guarantee that you get the disease. It's a risk factor. So these are complex genetic disorders, and I'm going to move swiftly on because that's not what we're talking about today. What we're dealing with in the NTSAD diseases are what's called monogenic diseases, which means a single gene disorder. And very inappropriately, these are called simple. So they are simple because only one gene is responsible. But from the minute that gene is dysfunctional, there is nothing simple about these diseases. But what I'm hoping to convince you is that the complexity of what goes wrong gives us lots of clinical opportunities to treat different aspects of the disease. So although it's complicated, that complexity does have some applications to the development of new treatments. So they're single gene defects. Most of them have a simple pattern of inheritance, typically what's called an autosomal recessive disease, where both parents have to carry a mutant form of the gene to pass it on to the children. And that's a probability of 1 in 4 pregnancies being affected. Now, the other thing to bear in mind is, although these are simple gene defects, we know that the rest of your genetics is a modifier. So there are other genes and other aspects of your environment and your diet that may make the presentation of the disease different even between siblings. So although this is simple, it's simple in inverted commas. So this is just really highlighting the fact that we have this single gene defect, and that's why gene therapy is one of the very attractive approaches, because the idea is to replace that faulty gene copy with a fully functional one. So that's the aim, and that's going to be the panel discussion. So Tay-Sachs and allied diseases, they're simple genetic monogenic diseases, as we described. They're also classified within the scientific and medical community as metabolic diseases. They're called inborn errors of metabolism. And I'm not sure what that means to people. It's a kind of a strange term. And metabolism is everything. We sort of think about metabolism as we eat food and we metabolize it and we have energy. And if we don't eat enough, we're too thin. And if we eat too much, we may have a tendency to overweight. And we sort of think about metabolism in that sense. But metabolism is going on in every single cell of our body all of the time. And it's incredibly complicated. And I'm about to show you the reason that I didn't do biochemistry as an undergraduate degree, because I was put off by cellular metabolism. So can you sympathise with why I became a zoologist? It was much more tangible to work on insect physiology. The metabolism of our cells was worked out a lot in the 1940s, 50s, 60s. Absolutely fundamental. And there are fashions in science. We have things that are very topical to work on, and there's money to work on them. And metabolism became the poor relation. It was unglamorous, it was mundane, we knew all of the players, there were all of these endless enzyme reactions and metabolites that happened in different bits of our cells. But I'm pleased to say it's undergoing a renaissance. And that renaissance in the interest in metabolism is actually going to pay dividend for treating these diseases, and I'm going to explain why in a moment. I'd like to simplify metabolism down to the London Underground map. Has anybody been on raise your hands if you've been on the London Underground? Okay. So it's this stylized map of London. And it's just to illustrate the fact that if you imagine that this is the metabolism of our cells, and we have a mutation that affects, for instance, this little bit of the northern line, you can imagine that the impact of that for most of cellular metabolism is actually quite minimal. So if you had a drug that would fix the ability to go from Tooting Bar to whatever the end of the Morden, that would fix that disease. But if you have a disease that affects the line that goes out to Heathrow Airport, although the rest of this network is okay, you can see that this is beginning to cause serious problems. So for transport machinery in the cells, this would not be good. But what's even worse is if you knock out something like King's Cross St Pancras Station, because that's the Eurostar line, that's the hub for this whole central network for inner London. And what I'm showing you this for is that if you could find a way of mitigating the effects of any one of these problems in our cells through these different genetic diseases, you don't necessarily have to fix the whole problem to have benefit. So I like to also illustrate it like this, that we've got our gene defect and it leads to all of this metabolic flux and changes within the cell. And this actually looks fairly straightforward because you've got sort of a problem at the top and you've got this storage down here of all the water. But the reality is that this is what we're dealing with. And what we really need to do is chip away at understanding all of these different little aspects. Because if we can prevent this side of this from happening, that would probably have a major clinical benefit in patients. So this is the sort of philosophy behind how some of the treatment approaches are being developed at the moment. Another way of looking at this is to sort of put the cascade into a schematic and say we've got a gene defect that we know is the cause of the disease. So obviously, if you could fix that, that would have the most clinical impact, at least in principle. We know that that gene codes for a protein, typically an enzyme. If you could fix the protein by giving it intravenously or using a bone marrow transplant approach or using a small molecule to enhance its activity, or to try and get more of this protein made by the cell and folded properly so that it's functional, that could also have benefit. Now you can see that this is an expanding list of approaches just for fixing the protein. The other thing is that we know that things are stored, lipids are stored in these diseases. If you prevent the lipids from being stored by using a small molecule drug called a substrate reduction therapy drug, that is also an approach. So this is a small molecule based approach, and downstream of this, many of these approaches are now using small molecule drugs, not biological therapies like enzyme replacement or gene therapy. So we have this very complex cascade, many therapeutic intervention points still to identify, and also I should highlight that you can also use stem cells, or at least in animal models you can use stem cells that will either supply enzyme or fix the bridge between cells that have lost their connections. So the thing to understand is, yes, it's complicated, but look at the number of possible ways that we could intervene. And the challenge now is getting those approaches actually into clinical reality. So this is a slide that I've been very kindly given by Sue, which shows you the diseases that we're talking about, Canavan, GM1, GM2, and we're looking at the ways that you can look at the gene that's affected that we know, the enzyme that's affected, we know what's stored. And this is just to illustrate that some of the approaches that are being taken at the moment are to use things like gene therapy. If you can't fix the gene, can you replace the enzyme? And if you can't do either of those things, can you reduce the buildup of substrate? And none of these are mutually exclusive. If you were to combine all of these approaches, you'd have a much, much better outcome. So it's not either or, it's actually all of the above. And I think that's the other take-home message, is that at the 70th anniversary of this foundation, we'll be looking at combination therapy results in the clinic in this patient community. It's not going to be a single treatment. Now, in terms of gene therapy, because we're about to have a panel discussion, the gene is made of DNA, and you can't just take a piece of DNA and inject it into somebody, it would just get broken down. So you've got to come up with a way of delivering the gene to the cell so that it actually integrates and actually does its job. And the idea is that you introduce on top of the dysfunctional gene a functional copy, and that will act as a source of the enzyme. Now, paradoxically, we do this by harnessing what we normally think of as bad things, which are viruses. So you can make a virus safe so that it doesn't give you a disease, and you can package the DNA for the gene in question into a viral vector. So it's basically a safer virus that goes and does what a virus does, and it actually is a very, very useful biological, very efficient way of delivering genes into cells. And you'll hear about two different sorts, AAV and lentivirus. They're two just different biological virus systems that can all be used. We can exploit them to deliver genes for us, and you'll hear about that more. Now, the other thing that's complicated about gene therapy is that there's two ways of doing it. You can either give it directly to the person in vivo, which means that you would be injected with the virus, either directly into the brain or into the periphery, and that virus would then go and deliver the gene to the different cell types that are affected. The other approach is called ex vivo, which just means outside the body, and there you would take cells like blood cells, for example, and you would put the virus into those cells in a dish in the laboratory, and then give those same patient cells back to them so that, for example, their blood cells can be a source of the missing enzyme. So when you hear about in vivo and ex vivo, these are two different approaches. From a practical point of view, you need a huge amount of virus to do this, particularly in adults, whereas you need very little virus to do that, which is the sort of practical consideration. When we're talking about enzyme replacement therapy, which is used for several other lysosomal diseases, this has to be given intravenously because this is a big protein and it will be broken down if you were to take it orally. And also for CNS diseases of the brain like the ones that we're talking about at NTSAD, this may have to be given directly into the brain itself, either through a pump or through intrathecal injections, which is into your spinal fluid. So this is complicated, and the reason that you have to give this directly to the brain is that these proteins can't cross from the blood into the brain because there's a thing called the blood-brain barrier. So one of the big challenges in all lysosomal diseases affecting the brain is how to get any therapy across from the periphery into the brain. Now, you've also heard about stem cells. What are stem cells? These are cells that can basically divide and replicate themselves. We have them in our bone marrow. They make all of our blood cells all of the time. We have some in our nervous system in the brain, particularly the bit to do with our sense of smell. And they can be driven to differentiate into all sorts of different cell types, muscle, brain cells, liver cells. And so you can harvest these stem cells to either do a transplant of them where they're taken from a healthy donor so that they've got fully functional gene and protein, or you can take the patient's cells and engineer them with these viruses to have the proper gene and protein product. So this is something else that you may hear about, and that's what their basis is. Now, my interest is in small molecules. So this is what can you do with conventional drugs that may be useful in these diseases. And at the moment, substrate reduction therapy is the only approved drug. And there are two drugs. There's miglustat that we originally developed for Gaucher and for Niemann-Pick C. But there's also a new Genzyme product called iliglustat, which is another substrate reduction therapy, small molecule for Gaucher disease. So these are things that are in the background. They may be disease modifiers, and those are something to just be aware of. They reduce the buildup of storage in cells. Chaperone therapies are another interesting set of small molecules. They will basically try and get the most activity out of the dysfunctional enzyme by augmenting its activity. So these are small molecules that bind to the enzyme and make it more capable of doing whatever that enzyme is supposed to do. This is substrate reduction therapy, just to illustrate the fact that if you're making molecules and you can degrade them properly, you don't build up anything. So the flow of the tap is matched by the size of the plug hole, if you like. So if you imagine this is a bath, you run the bath in the hotel. If you leave the tap open and your tap is flowing at a certain rate, you don't flood the room underneath your particular room. I'm in 515. Just whoever's in 615, heed this. Yes. So all those warnings about having the shower curtain inside your bath become very relevant. Now, in a storage disease, the molecules coming into the lysosome to be degraded carries on unabated, but the problem is that we're not degrading things. So you can see that the risk of flooding is really very high. And all that substrate reduction therapy is doing is turning down the tap so that the drips that go in are the drips that come out, and that's really the principle of it. The chaperone therapies aim to stabilize mutant enzyme. Pyrimethamine has shown activity in GM2 storage disease cells, and there's some clinical data. And I just want to mention another drug that's in clinical trials in Europe at the moment, a thing called arimoclomol, which is a drug, a small molecule that you can take orally. And it's a protein that enhances the cell's normal stress responses, and it upregulates a protein called HSP70. And it's in clinical trials at the moment for Niemann-Pick C disease, but also it's going into trials in neuronopathic forms of Gaucher disease. And in this paper, Thomas Kierkegaard and colleagues and James Gray from my lab showed that, in fact, if you take a panel of different lysosomal storage diseases, you can correct them in tissue culture with this drug. And I'm just going to show you some data from a mouse model of Niemann-Pick C that we've treated with this drug in my lab. And this is the back part of the brain of a mouse model of Niemann-Pick C, the cerebellum. This is a normal mouse, showing the green is myelin. This is the membrane that insulates all of our neurons and allows the nervous system to work. It's a bit like insulating tape. I guess if you wrapped that round electrical wires, that's the kind of thing that myelin is. You need it for your nervous system to work properly. This is the situation in Niemann-Pick C. You can see a very, very small amount of green compared to normal. If you put the protein in that we think is the thing that's modifying the disease, HSP70 as a if you like, it's like enzyme replacement therapy. This is not an enzyme, it's a chaperone. But if you put the protein in, you can see that that looks much better. This small molecule oral drug, you can see this really, really nice retention of myelin. So this is a watch this space kind of area, because if the trial is positive in Niemann-Pick C, it will be then taken forward into clinical trials for other lysosomal diseases. It has two mechanisms of action. It improves lysosomal stability, which is the only thing that we're fixing in this mouse model, because it has no protein, it doesn't make the NPC1 protein, but it also will enhance protein levels and activity. So it's going to be very interesting to see the outcome of clinical trials. Those are going on at the moment in Europe and also with a small site in the US. So what does the future look like? Well, I think it's almost certainly going to be combination therapies. And in addition to the things that I've talked about today, there'll be many, many others that come through these seed awards from NTSAD. People having bright ideas about something that might modify the disease. The big challenge is, how do we effectively deliver to the brain? This is the vasculature of the brain, and it unfortunately has this barrier that is biologically very important. It protects our brain and is actually a very good thing, but it is a barrier for therapeutics. So one of the big things is, how do we deliver things to the brain in as safe and as non-invasive way as possible? And some of these small molecules that cross naturally from the blood into the brain may be very, very useful in this respect. So I'll finish there, and then we'll move on to the panel discussion. Okay, so we're now going to move on to a discussion focused only on gene therapy, to give you an update of where this technology is, where the translational potential is. And we have a very esteemed panel, and I'm going to ask you if in turn you could just introduce yourself and say what you work on.
Okay, so my name is Dominic. I'm from UMass Medical School. I'm working in Guangping Gao's lab and leading the Canavan research program there. And we are using AAV gene therapy to treat Canavan disease. And our main focus is to use a one single shot intravenously to treat the brain, basically.
My name is Joe Anderson. I'm from University of California, Davis in Sacramento. And our major focus for lysosomal storage diseases is Tay-Sachs and Sandoff right now, using lentiviruses and genetically modifying the immune system. And based off of those results, we've also recently started work on GM1 and Canavan as well.
So I'm Doug Martin from Auburn University College of Veterinary Medicine. We have animal models of GM1 and GM2 gangliosidosis, cats and sheep. So just so everybody's clear, Dr. Heather Edwards is now in charge of the sheep. Heather, please stand up, wave. So any questions about the sheep, please direct them to Heather. Also want to introduce Cassie Bebout. Cassie, please stand up. She's an undergraduate student from my lab whose brother died of GM1 several years ago, and she has now come to the lab with the goal of doing a PhD and doing lysosomal disease research. So we're really glad to have Cassie here too.
My name is Sena-Esteves. I'm from the University of Massachusetts Medical School, Gene Therapy Center. I've been working for quite a few years on AAV gene therapy for GM1 gangliosidosis and Tay-Sachs disease, and I think I know quite a few people in this room. So I think I'll leave it at that for now.
Okay. Well, thank you very much. You can see a very esteemed panel. And the way this is going to work is quite flexible. That we're going to start by asking, I'm going to ask a number, or bring up a number of issues that may be of interest for understanding gene therapy. But we are going to then throw it open for questions. And also, if you have a question during any of this interaction, please raise your hand, and we're very happy to take questions from the floor during this, not just at the end of the session. So I'm going to open this up to the panel to discuss why it is that when we've got so much promising data showing that gene therapy works in mouse models, that we're not already in the clinic. Could somebody comment on what are the steps from mouse to clinic that may not be obvious as to why there are steps in between and why they're so important?
A nice, easy question to open it up. Well, yes, since I made this smart aleck comment, I guess I have to respond. There are lots of steps between going from showing that something works in a mouse and showing that it's possible that it could work in people. And so for instance, a mouse brain is about 2,000 times smaller than the brain of a child. And so something that works in a small mouse brain is not necessarily going to work in the large brain of a child. So that's one of the things that you have to deal with, is the scale up. Another thing that you have to show is safety. You can't just show that it works in mice and cats. You have to show that it's going to be safe, because the FDA, their first job is to make sure that we're not doing clinical trials with things that are unsafe and that can cause harm. I'm going to let the other guys talk, but there are lots of regulations and lots of paperwork that go into the whole trial approval process. And Dr. Tiff will talk later. She probably can address some of that as well, but I'll pass it off to my esteemed colleagues.
Well, I can just agree what Doug just said. Another issue is, for example, Kahneman disease. We have a lot of good mouse models, but there's really no large animal model, like either in a cat, dog, monkey. So obviously you could just treat a monkey to kind of bridge this step from mouse to a human-enabling study, and you could treat a monkey that's a healthy monkey, but that might tell you something about safety, but you don't really know necessarily how well the therapy might work in a larger animal model. So one approach we have taken is we are creating a Kahneman monkey, but the problem is the monkey we are using, they're fertile after 5 years. So you can imagine, and they live 16, 20 years. So you have all of a sudden, instead of 3 weeks breeding for a mouse, we're talking here about 5 years breeding a monkey. And that expands obviously the time it will take to have the appropriate large animal model if it's not available. That definitely makes it more difficult and stretches the transition quite a bit.
So I could make some comments on that. I think the field is also changing very rapidly. As we gain, more and more clinical trials are being carried out, which tells us a lot about safety in humans, right? So the barrier and the timing between doing experiments in mouse and translating them into human is becoming shorter and shorter the more we know of how these entities perform in human, right? Many of you, we unfortunately had an example of mice might not be predictable of what could happen in a human. In 2012, we decided to do a monkey study, essentially to check a box for us, really. Actually, it was not mandated by the FDA at all. We thought, well, we're putting in, for the first time in a human, in a structure that no one has ever done before, a virus that a particular one that no one had put in a human. So we thought, maybe we should try this in a monkey. And this is after doing experiments in mice, cats, and sheep. All of them safe. All of them reproduced the biology that we had first discovered in mice. And I call that the worst and the best decision we've ever made, okay? The worst because, well, obviously we're here, so we haven't started a clinical trial yet. The best because what we found in monkeys would have been a tragedy finding that in human, okay? It tells you a little bit that it's a little bit unpredictable to a certain extent. What you do in a mouse may be reproduced in human or not. And at this point, actually, until we do a clinical trial, we really have no idea which species to believe. So we did the experiments in monkeys, we found horrible toxicity, essentially stopped the program, and we had to rework things quite a bit. Fortunately enough, we're now in a position where we actually have fixed the problems and in a great position to actually move towards the clinical trial. Had we not done that, we could have not only stopped our trials for good, we could have affected the entire field, okay? So 2000, some of you may have heard that a kid died at UPenn in a clinical trial. That came as close as it ever did to killing a field, okay? It almost killed the gene therapy field. It's again one of those events that was incredibly unfortunate, but it was also a great event, which was to teach the field that we have to become more rational on what we use for each disease to be the appropriate entity for each disease. So the field overall has become much more rational out of that. So out of events that really are not pleasant, I mean, a human is a tragedy, a monkey is bad, we learn something, okay? And what we learn is from our experience is that you can take absolutely for granted that just because it's safe in a mouse, it's going to be safe in a human. So when you do this jump, right, you try to do it with as much information as you can, right? Because, you know, you have a problem with a mouse, okay, it's unfortunate, delays the experiments, etc. You have a problem with a cat, pretty upsetting actually. You have a problem with a monkey, really upsetting. You have a problem with a human, that's beyond upsetting, okay? So that's why it takes a long time to go from, okay, oh my God, look at these wonderful results in a mouse, right, to go into a clinical trial, because we want to make sure that we've done everything possible to show that it's actually safe, or that there is it's reasonably safe, that we're pretty sure it will be safe, right? The worst thing that we can do that's going to set back everyone is to go into a human trial and have horrendous toxicity. Right? So that's why it takes a long time. Now, on the other hand, as I said before, as the field is progressing very rapidly and gaining more and more and more knowledge about the safety, the time between getting that critical result of showing, oh my God, this strategy actually does work, it's going to become shorter and shorter and shorter, because all of us have gained the experience now in humans to know that, in fact, it's safe. Okay, so hopefully what this will become, and not just for Tay-Sachs, for GM1, for Canavan, and all the other plethora of these rare diseases, is that we are probably going to get to a situation of plug and play, where it's going to become almost like Lego, right? You just take the red piece, put the blue one, and yeah, it still does the same kind of thing, right? That's where we hope to be. We're not quite there yet, but that's really where we hope to be, and I think that the evidence that's starting to suggest is that we will get there, right? Because this is accelerating very rapidly, and the safety profile is really being reproduced from one disease to another, to another, to another. So that's fantastic news, but to get there, it has taken a long, long time between getting those fantastic results in a mouse to getting to a human. And so the time frame is going to become shorter and shorter and shorter. So that's, I mean, why it has taken a long time for a lot of us to go from that first result to the human application. At least that's my perspective.
Yeah, and I agree with everyone else on the panel as far as taking the jump from mice to humans. And with our experience with our HIV trial that we currently have now, we were able to go from mice to humans, and we didn't have to do any large animal model. And a lot of the times that's up to the FDA. In similar trials like our HIV trial, other groups have had to use large animal models such as non-human primates to do the studies to look at safety, but for ours we didn't. So our method is a little bit different than my colleagues' AAV, where we are using an integrating vector that's going to put the gene into the targeted cell's chromosome so it's there forever. And we're not doing a brain injection or an IV injection. It's actually an ex vivo gene therapy method, like Fran had talked about, where we genetically modify the cells outside of the body. So we're not injecting a drug or a virus into the body. We're actually doing the genetic modification outside of the body. And we are regulated by the FDA to only allow 1 to 2 of those copies into the cell. So as far as a dose regulation of a virus or a drug, we are actually contained by the FDA to only put 1 to 2 copies of those extra genes into the cell. And then the difference between our approach is that when we use the blood system to express this new wild-type form of the protein, it's going to be expressed throughout the body in places that it might not be expressed normally, because the blood flows throughout the body, goes to almost all, if not all, the tissues and the cells in the body. So that's one thing that's going to be different between our methods. So we do have to look at a lot of other safety measures, because these overexpressed proteins are going to be throughout the body, not just in the brain, but all over. So that might be an issue with the FDA where we may need to do a larger animal model as far as the safety goes. As far as efficacy, there's no, I shouldn't say there's no larger animal models, but as far as non-human primates, it may just be a safety experiment that would have to be done in the monkeys and not so much efficacy, but that will also take more time. Did you have a question?
Okay, so I understand the changes done outside. Oh, sorry. So I understand the changes done outside of the body, but then you're putting back, I would assume, a volume of blood, and probably even large volumes of blood, so that it's going throughout the whole body. So why is it safe to put back a large volume of blood that's going to throw throughout the body and affect everything, but, well, just that, why is that considered safe and not needing further testing along the way?
So we actually don't put the entire blood volume. We first isolate the blood stem cells, which in the periphery, they circulate. They're about 0.01% of the actual cells that circulate. And then in the bone marrow, there are a larger number of them. But we isolate the stem cells first, genetically modify them, and then put them back into the body IV. And they'll actually home back into the bone marrow. They don't circulate throughout the body for the life of the patient. They'll home back into the bone marrow because they have that direction to go. And then throughout the life of the patient, they'll produce more immune cells like T cells and B cells, which will be genetically modified to then secrete those proteins throughout the body. You understand?
Yeah, but if there's a problem, how do you call them back?
Call them back.
So that they're not creating problems throughout the body. I understand it's going just to the.
Well, that's what we would initially test in the preclinical models to see if that enzyme secretion and that overexpression of that enzyme is actually doing some sort of negative effect to other parts of the body.
I think the point that you're raising is a very good one, and that is that if you take a drug and you have an adverse effect or a side effect, and it's something that really affects your quality of life, you can stop taking the drug, and over time it's eliminated from the body. With these biological therapies, once you've done it, it's very difficult to go back, and that's why there's so much concern about thinking through every conceivable thing that may go wrong and trying to make sure that it doesn't before we go into people. And I think actually that brings us very nicely onto another question, and that is when people talk about safety, what is safety in real terms, and how do you define the limits of tolerable safety? What's the cost benefit and who makes that decision? Could you comment on that?
Sure.
Go for it.
I can go first since we were talking about the blood system. So as far as safety, since we're looking at a specific stem cell, the first thing that we want to see is, does that stem cell act the way it's supposed to act? Does it make the cells that it's supposed to make in the body? And then do those cells function properly? Because we don't want to genetically modify a stem cell, do a transplant in a patient, and then not have the cells engraft, not have them make a normal functioning immune system, where then the patient becomes immunodeficient. So initially we look at the stem cell function and differentiation and functionality of the end stage mature immune cells first. And then we look at the other parts of the body or the other organs or in the brain tissue, and to see if those look normal, the right sizes of the cells, the right sizes of the tissue, the normal function of the brain. So those are all the experiments that we do as far as looking at the safety of our gene-modified cells. And then ultimately it's up to the FDA. Do they say, yes, you have done enough experiments, does it look safe enough? Do your gene-modified cells look the same as non-gene modified cells? And in grafted mice, transplanted mice, or animal models look the same as non-transplanted animal models?
So, Joe, Florian, I assume you're using myeloablation, or you're planning to do that in humans, similar to what is done in transplantation?
They might need an explanation of
Right.
myeloablation.
I thought, making room in bone marrow, and maybe also talking about long-term safety of insertional mutagenesis. I'll leave that to you.
Yes, so his question was, are we planning on doing myeloablation, which is a complete ablation or knockout of your current bone marrow and immune system, so that most of the cells that we genetically modify and engraft will be those cells that go in. So right now we're working with our bone marrow transplant doctor at UC Davis who can do juvenile and adult patients. We don't have a pediatric transplant division at UC Davis, but we're working with a couple other pediatric transplant doctors at UCSF, so San Francisco, and then UCLA, who are writing up those protocols. We are thinking of doing a mild ablation at first, just for the phase 1. So that means we wouldn't do a complete knockout of the immune system. It would be a mild knockout of the immune system, so that we can look for engraftment of the gene-modified cells, look for safety of these cells, make sure that we're not doing any harm in the human if we do that jump from mouse to human, if the FDA allows us. So it won't be that all of the cells in the body are gene-modified. There will still be some that are not, and because it's a mild ablation, it's possible that we could take some cells from the body and freeze them down as non-gene modified, so that if we do see any problem with the gene-modified stem cells, we have this backup product, so we could then go to a full ablation of the immune system, put this non-gene modified backup product back in, just like a normal bone marrow transplant that's done with cancer patients. So that is an option, and we have that option of a safety backup of these non-gene modified cells, so we can ablate the gene modified ones and then put back in the immune system that the person had before the gene modification.
Heather,
Okay.
well, just to kind of step back, because there's scientists in the room and researchers, but the parents and families. So as you're going through, and sorry, Heather Lau from NYU, as you're going through, that was helpful to explain each platform that you're using, because there's different approaches to gene therapy. So as you're talking about safety, that question that you had posed, can you just preface it like Dr. Anderson just did with your platform that you're using? So in this case is immunosuppression or myeloablation, and then gene therapy to blood cells, but you all have different approaches, correct? So as you go through your safety discussion, can you just give us a little brief one-liner of how you're doing your gene therapy?
Well, well, in general, it all comes down to your immune system, more or less. And for the AV field, at least, I guess there are two major aspects of your immune system we are looking at. One is obviously we're using a virus to deliver the healthy genes. So the virus itself is basically, it's like the envelope of the therapy, but the envelope, like if you get a cold in winter, right, that's caused by a virus, you get all those symptoms because your immune system responds to it. In a similar way, AV doesn't give you fever or anything like this, but your immune system might still recognize it as something foreign or that doesn't belong to it, and so it will respond with the immune response. The other aspect of the therapy is if you have a mutation of a gene and you have no expression, so that you have no protein at all from this gene. If you reintroduce the healthy gene and then you get a normal protein, your body has never seen that protein before in his life. So the immune system also looks at it, even if you're supposed to have it, but your immune system will still look at it as a foreign thing and might respond with an immune response, which can cause a lot of problems. And obviously there are many ways how you can look at the immune system and its response. But then the question is, obviously, if you know there might be an immune response, how to avoid in a patient. And at least right now the field is trying to use immune suppressant drugs that are already available and used for other diseases, like all these inflammatory diseases of joints and stuff. And so you can use those drugs to immune suppress the patient for a certain amount of time to avoid any form of severe immune response. You can do this prophylactically, or you can treat the patient, wait until you see like a spike in certain marker we look at usually, and then you start treating the patient. So there are different ways of doing this. But yeah, so those are the two, I guess, issues in the AV field. I'm sure you have something to add.
Oh, you look at me. Okay.
Well, I look at both of you.
I suppose. So I think that the simplest definition of safety is that do no harm. Right? I guess that's the simplest definition. And in the case of one of the important things to consider in all of this, as Fran has mentioned, is there is no point of return. I mean, once you've triggered it, if things go wrong, they will continue to go wrong. So you'd better look at it very, very, very carefully. And certainly how the immune system will look at the new enzyme that you're making is absolutely critical. Because fortunately enough, we've learned, for many years from, transplantation that, yeah, we can actually control the immune system to a great extent. But that's, you have to take into account the fact that it is a permanent modification. Especially when we're talking about AAV, direct injection into the brain, or a delivery through the bloodstream. Those viruses, fortunately enough, are going to stay there for the lifetime. And our experience is that not only they stay there for the lifetime, is that they express and make the normal protein for the lifetime. That's of course fantastic. That's what we want. But that, has to be used very carefully and fully conscious about the potential consequences of it, right? So the simplest definition is that, for instance, you don't kill cells, right? I mean, if you inject one of these viruses into the brain, is that you don't induce cell loss. I mean, you don't want to make things worse. So you're going into a brain that potentially is already sensitive, has a disease process, and the last thing you want to do is accelerate it, right? So the definition to a certain extent is simple, right, at the surface. Now, if you want to dig deeper, I'm pretty sure that you will find things that are altered, right? Because you're asking cells to do something that they didn't evolve to do, right? And I mean, that's what Joe was saying, right? All of this, if you think about it, is you're asking cells to do something that's not how we evolved, right? Every single one of our cells makes a certain amount, right? The entire principle of therapy for the lysosomal storage diseases is that you are engineering cells to make a boatload of it. Okay, so if you think about that, that's not how your cells evolved, right? So it's quite possible that if you look deep enough, that you'll see that the physiology of those cells is altered. The question is it altered in a way that results in them dying, or is it altered in a way that, okay, fine, they can do that too, right? What we found in our monkey studies that we asked maybe a little too much out of them, okay? But the reality is that if you have, over a long period of time, that you're asking these cells to do something that is physiologically abnormal, and I think we all realize that it is abnormal, if it's compatible with survival of those cells and not cause dysfunction and have a therapeutic effect, to me, that's a good definition of safety. As a scientist, we can go look deeper, and I'm sure we will find the signs of that altered physiology. Okay.
I would like to comment on some of the issues that were raised by the panel and could be useful also for the for the families. My name is Sandra Dazzo. I work on lysosomal diseases for, I think, about 40 years. And I work on a group of diseases.
Can I interrupt just for a second? So just for you to understand, Sandy has pretty much characterized the entire GM1 field. Okay, so I have to, no. Believe me, I grew up reading your papers, right? From sort of characterizing the biochemistry. So just to give you that background, that it's really important, is that Sandy has done more than anyone else to understand how this disease works, okay? So this is, those 40 years is with that background. So I think it's important that you understand who you have. So it's important, right? Because all we can do, right, is because of the work you did for 40 years, right? No, it is true. That is the case. So that's important to recognize that. So sorry to interrupt you.
Thank you very much. Thank you. What I actually want to share with you is the experience I personally have with families and patients who are contacting me directly to ask what is the next step and when these are going to be effective, when things are going to happen. And it's excruciating for scientists like me. I'm not a clinician. I have been obviously sharing with the families their hurdles and difficulties and sorrow. But, and I'm trying to do my best to actually address the very, very fundamental issue that actually Fran very nicely, and of course Miguel and Doug and
Joe
and
Dominic.
have been addressing as well at different levels. We are really have to balance the time, because on one side, and I see it, I work at St. Jude Children's Research Hospital, I see how the clinicians address treatment of cancer pediatric patients. And there is an absolute urge and need of finding something to do for the children, considering that, of course, the diseases, starting with lysosomal diseases, are progressive, so obviously they continue to deteriorate. So on one side, you want to do something fast and as fast as possible. On the other side, I think we really need to understand, and I cannot say it enough, and I think this addresses also safety, toxicity, whatever the panel had been discussing, immune response, is the fact that we don't really, unfortunately, as Fran very nicely said, these diseases are complex and are really complex. Often enough, we tended in the early days to actually generalize. You can use one approach for all of these diseases. It's not as simple. Some of the approaches can be used for multiple diseases. Some others don't respond at all. So maybe, as Franz said, the possibility to use chaperones that might help in multiple diseases is very attractive. On the other side, I don't think we know enough what these enzymes do and what their metabolites do. So often enough, we tend to address exclusively the fact that if you give a little bit more of the enzyme, you obviously eliminate some of the byproducts or the storage products, and that is enough. Sometimes is enough, sometimes is not. You don't know what is happening at the cellular level. You don't know what else is downstream of these metabolites that might be initiated as a standard pathway, and that is obviously the hurdle that need to be considered. I work on a complex of 3 enzymes, one of which is beta-galactosidase. As Miguel very nicely, again, and eloquently said, we don't know really if you, and I discussed this with Doug and Gorey and Cindy last night, we don't know what to do, what we do when we boost the enzyme, one of the 3 enzymes, to the limit. What happened to the other 2 that interact with this enzyme? So we are really, I think the understanding of the field is definitely moving. And we are in a very, very good time to team up, figure out how to work together to understand better how to tackle it and to tackle the diseases. So I think it's really better to address with cautiousness the generalization. If we find something that we can generalize and use for multiple disease, perfect, but be very, very aware of what is happening at the cellular level. And most importantly, of course, we are talking, Fran talked about the neurons, kidney cells, liver cells, hematopoietic cells, what happened at the cellular level at all these sites is important to know. So has to be done really in parallel, and hopefully the field is moving in this direction. I don't say that we shouldn't try and address this. I have another point that actually I was wondering about, and maybe I have a way to get the comments from you guys. Sometimes, of course, the very, very big issue is toxicity. We were talking about it last night. So toxicity, we use monkeys to actually try to see if the closest, obviously, primate to the human. So in theory, it should be a good way to actually potentially test how toxic the therapeutic product is. However, you use the vast majority of the cases monkeys that are normal, right? So they have already a basal level of enzyme. They have already regulated levels of this enzyme in all the different sites and cell types. So what happens when you just overload the system with a gene therapy approach or enzyme replacement in an animal that in fact is a wild type, is a normal animal, right? So because I still think that a model of a lysosomal disease that mimic the lysosomal disease is still much more informative because they might respond very different.
Yeah, that's a very good point.
So
thanks, Sandy. That was an extremely insightful set of comments. Do you want to just join the panel for a second? Because what I'd like to do now is open it up to questions from all of you. What questions from what you've heard this morning would you like more information on? Yes.
I wonder if you guys could speak to the current state of vector manufacturing. It seems like it's backed up. It seems like it's incredibly expensive. What's the story behind that?
In the interest of time, could one person comment on each of the questions from the floor so that we can get as many questions as possible?
Yeah, it's an issue that a lot of people are running into. There are a lot of good preclinical results in animal models, and people would like to move on to try to get clinical trials done. As Fran mentioned in her setup, direct injection of gene therapy into the bloodstream, for example, requires a huge amount of vector. And there really aren't many facilities that can make vector at the purity and the level that you would have to have for a clinical trial. I think that's changing. A lot of people are seeing the utility of gene therapy and that it is coming into the fore, so a lot of companies are investing in their own gene therapy manufacturing facilities. But you're right, it's a bottleneck right now. And For example, say a typical early phase clinical trial that has 4 to 8 patients, you're looking at somewhere around $750,000 to make enough vector to treat those kids by the IV route. So it's very expensive.
Yeah, so the manufacturing is still evolving, right? And I think we'll get to a stage where it may become cheaper, but I think we're still early on that the manufacturing capacity has not reached a level where it starts to push the prices down. So I think it's still at the early stages of a technology where prices are still pretty high. And right now, there is a manufacturing need that is not yet met, throughout the country actually, and I would say even throughout the world, as a matter of fact. So yeah, that is definitely a bottleneck for the entire field. And there are companies that are expanding very, very rapidly to try to fill that gap.
That's great, thank you very much. More questions, good.
You've mentioned the FDA. So the president mentioned it in his address to Congress, and I'm skeptical of him because I think he doesn't do a good job of addressing complex subjects well in terms of his simplistic solutions, but I try to be open-minded here. So objectively, are there ways to improve FDA processes and procedures?
Aside from the political entertainment,
I know.
which one could have an entire session of just fun points, I suppose. Our interaction with the FDA has actually been very, very positive, and we tend to think of them as a block, as a roadblock. My interactions with them have changed a little bit my opinion on it, which, the interactions that they've had with us is as colleagues that have a heck of a lot more experience than we do, because they've seen way, way, way more than any of us combined in a single room, okay? They've seen horrible things, they've seen things that no one else knows, aside from the people that are running them. And these guys see the entire spectrum, right? So our interactions with them have always been very positive. They actually have given us great guidance on what to do to get to the clinical trials. So in all honesty, for us, I don't think they've been a roadblock. And in fact, the latest interaction we've had with them was incredibly positive. Actually get to the sense that these guys actually want to see these therapies for rare disease go forward as fast as possible. Yeah. But with that mandate, their mandate is do no harm. After all, we created the FDA, or the FDA was created because of snake's oil salesmen, right? That's why it was created, right? So that we're not putting into people the snake's oil that really is not going to do anything. When people, it's easy to blame the FDA, right? It's that big target that it's really easy to go after. And I think you hear this pushback about the FDA simply because I think often companies want to move things really, really, really fast. And the FDA kind of pulls them back a little bit. And I think there is that push and pull between the safety and getting things fast to the clinic. Our experience for gene therapy, and I can only speak about that because I don't know anything else about them, has been incredibly positive, and I believe that they actually want these things to move to the clinic as fast as possible. So I don't see them, for us, at least, as an impediment, really, to moving forward. I really don't, actually. But that's just one sliver, very, very, very thin sliver of, of the view of the FDA. So, but that's my relative inexperience when interacting with them.
We've got another question at the back, I think. Yeah.
Hi, I'm from Biomarin, and my name's Gori, and my question is for Doug and for Miguel. I just want to follow up on Sandra's comment about the toxicity potentially seen with gene therapy, and what I'd really like to see in the papers coming out for gene therapy, particularly for GM1 gangliosidosis, is more evaluation of the multi-enzyme complex. I think it's really important to make sure that other members of this multi-enzyme complex aren't being affected when you're coming in with a huge excess of one of the lysosomal enzymes, such as beta-galactosidase. And the mouse and the cat studies look fantastic. They look very promising, and it would be great, to get an effective treatment for these children. But at the same time, the toxicity is important, and given that there is some toxicity occurring for related gene therapy studies in the GM2 model, I think it is important to really look at toxicity in the GM1 models as well. And I'd like to see in the next papers that come out some study and evaluation of these other components of the multi-enzyme complex.
Yes, so what Guri's talking about, for those of you who may not quite understand or follow it, is that beta-galactosidase exists in a complex with two other proteins, two other enzymes. And so the concern that she and Sandy raised is that overexpressing beta-galactosidase to try to treat GM1 could affect this so-called multi-enzyme complex because there's too much beta-gal, and it somehow might perturb that complex. I think we should be looking at toxicity from every angle possible. You know my argument, Guri, is that we have cats treated that are 6 years old now, and they normally would live only 8 months. So there may be some toxicity, and we certainly should look for it. And we will. Actually, we will start doing neuraminidase assays in our IV-treated cats. But if we can treat a child and have them live to 6 years when they would only live to 8 months, there may be that level of toxicity, but that's a big advance forward. So yes, we will look at that issue, and it is important.
So, if I may follow up on that, right? So it's again the risk benefit, right? We know exactly what happens if we do nothing. Well, that's what has been happening to these patients, well, I guess throughout human history. We know exactly what's going to happen. Also, I agree entirely that if you look under the hood, we are doing some things non-physiological. Absolutely, there's no question about that. But this is no different than any other drug that is on the market, okay? Every drug that is on the market, if you look under the hood carefully, you will find changes. There is no question about that. Now, the approach that companies take, right or wrong, is that they do not look under the hood, okay? And I can give you the example of, companies that look at RNAi, right? Silencing gene expression because some diseases are dominant and you want to kind of quiet them down, right? They do not look, they look at it as purely safety. Does it engage the target? Does it reduce the target? Is it safe? And the reason they don't look is they know that they open a Pandora box. It's a Pandora box that can last 20 years. Because if you know that there are changes, you're going to have to explain to the FDA, everyone else, what do those changes mean, okay? And the fact of the matter is that you can take the approach of saying, well, that's the scientist's approach, which is I want to understand what those changes mean. You can take 20 years doing that, or you can choose to say, look, it's safe, it addresses the disease, we'll take that for the time being, right? So it's sort of that balance between doing absolutely nothing until we know we can absolutely restore the genome, right? Because ultimately that's what we would like to do, right? Is go into the gene, flick the mutation, and that's it, off you go. You're normal. Actually, your genome is normal. Well, there's technology now that can actually give us that in a couple more years or decades or whatever it is. But in the meantime, we can choose to do nothing, or we can choose to do something, but with eyes wide open that, yes, there can be consequences, and you should look at it and be careful on what you do. But yeah, you're absolutely right. I mean, this is an abnormal physiology, without a question.
Thank you.
Just one quick thing, Fran. I think this has been a great discussion because maybe it gives you a little bit of a flavor of how complicated this toxicity and safety issue actually is. If I were in your position, I would say cats live in 6 years. How toxic could it be? But there are a whole set of different guidelines that have to be looked at when you're talking about safety and toxicity.
Yeah, and I think the other thing is that with all these treatments, you're converting one disease into a different form of the disease. Hopefully a much, much slower one, but it's still, and you may find things that emerge because people are living longer that still affect quality of life. So it's very, very complicated. And we've had a very interactive session. I hope it's been useful. I would like to ask for a family member to ask a question that's really relevant to them, please. There's a hand at the back to just close the session, because this is really for you, this session, not for those of us who work in the field.
So for the families, we're hearing a lot of the concern is safety, 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 but we're talking about overexpression of these enzymes. We're talking about injecting mass quantities. For us as families, can you explain why do you have to inject so much or cause an overexpression if that's the concern for safety? Why not regular levels like everybody else has?
Yeah, that's a good question. And
excellent question to finish on.
I can answer that, at least for cannabis, very simple. We don't overexpress it, so we reach roughly wild type level. So that concern, purely based on the overexpression causing toxicity is less of a concern for us. We are really looking more into, does it cause an immune response that's not related to overexpression? I think the overexpression really is more due to previous experience Doug and Miguel had.
Yeah, what we know is that we're not going to correct every cell. We're not going to put in a virus that transfects or infects every cell. And so to treat those cells that aren't infected by the gene therapy, there needs to be a certain level of overexpression from the cells that are infected so that that enzyme can be secreted and then taken up by the cells around it. There has to be, you need to hit the sweet spot. You can't underexpress it, you can't overexpress it too much. And fine-tuning that level is really the name of the game.
Okay, we've got one last question.
Thank you. Great discussion. My question is getting back to the practicality and to what is, let's break down, because it sounds like this is really complicated problem, many unknowns, toxicology, we can hardly know the experiment, the full model. So if we break it down, so what is known and what is unknown to you now? So we have this AAV pretty much knowing, maybe we have cases where it was used, so the transport is like, already we know this. So we can probably, and can you break down this thing, is what is missing for your perspective now until we move into the clinical? Thank you.
I guess I can address that. So, well, right now, in terms of, I can give you the example of the GM1 that is more relevant to you, that is the IV program that we're working on. We have a very well-defined program of experiments or steps that have to be taken, which are timelines are actually very well defined. We have in place contracts or proposals, cost proposals from entities that will do those experiments for us. So these are being done by what's called contract research organizations. So at this stage, the only, the magic ingredient that is mixing is the one that is always in the end boils down to that, it's money, okay? From the point of view of the interactions with FDA, we've had that. They agreed to the plan that we proposed to them. We have a very clearly defined timeline. We have clear activities. We have already identified who's going to do them. So at this point, at least for that program, it's actually, really, I would say for all the programs that you see up here, the steps really are a question more of having the financial resources to actually do them. Certainly is the what one could say the superficial approach that if nothing goes terribly wrong, then it's okay to move forward. We may not understand the full molecular details of what we're doing, but there is a reasonable expectation of an effect. Okay, I think let's take that also from all these safety concerns, is that the flip side of it is that there is a reasonable expectation of a success or of a benefit as well. So, but in detail, yeah, I mean, the steps are very well defined now. They're on very clear timelines. And should money not be an issue, it's certainly, there's no reason to believe that they would not follow such timelines, because I think of it as being in the glide path towards the clinical trial.
Okay, panelists. Don't get ready. We're going to thank you. We're going to transition. Only since I'm the next chair, I'm going to take the next chair prerogative, which is sort of the elephant in the room is the discussion we haven't had, which is our diseases are really severe diseases. And no one has really yet talked about balancing risk versus benefit. I mean, we know what the outcome is for our diseases, and it's not good. So I think even when the FDA begins to look at therapies, they say, what's the burden of the disease? How bad is this? Is there anything else out there? And the FDA is really willing to allow us to accept a little more risk in our experiments, because what's the alternative? We know what the alternative is. So this risk-benefit thing, I think, is something we haven't talked about, but is pretty important in the way the FDA looks at potential therapies. Yes, there are toxicities. Yes, there are things we have to worry about, but look at the diseases we're dealing with. So that's one piece. The second piece is, and we know this historically from other diseases, even other lysosomal diseases that we've treated, if we treat the thing that's the worst, and I guess the classic example is infantile Pompe disease. Kids with Pompe disease have these giant hearts, and usually about 3 or 4 months, the infants, basically go into heart failure, and they're dead certainly by 2, and often by 1 or 1.5 if you don't treat them. So they're uniformly lethal. So the enzyme therapy was developed by Genzyme. Basically, the first clinical trial outcome measure was, did these kids not die? I mean, that was the outcome measure. And the answer was they did not die. And this looked like the greatest thing since sliced bread. These kids were now able to sit and walk and run, and everything was great. Except that as they got a little older, we realized that, their muscles weren't working so well anymore, and they weren't speaking so well anymore. And so guess what? There are alternative things that came up with these kids that no one could have anticipated, because all the kids died. And so I think, we may run into the same kind of thing, is that we treat the thing that's the most important, and we see benefit, and we see really great benefit. But it's not to say that 5, 10, 15 years down the line, we're not going to see other things that come up. I mean, I think we've got adequate experience to know that that's actually what can happen. But I think at this point, trying to even look at first therapies for our diseases, we have to realize that the burden of the disease is huge, and that we may be willing to take more risk, and the FDA may be willing to allow us to take more risk in the therapies that they're willing to let us do. So, I mean, that's chair's prerogative for this one. So Sue also asked me to announce that she theoretically had programmed a break. She's worried that if we take a break, everybody's going to leave and not come back. So we're allowing people to sort of take breaks as they need to. Like, I got up twice because, I'm an old fart and have a small bladder. So feel free to take breaks as you need them. We're going to transition to something, sort of away from the science and more into what we've been talking about as a research group for a long time. As Kevin said, when we started this whole thing, 15 years ago, we were really interested in funding research projects that would begin to move us farther along. And we've been doing that for a long time. But now we're at the point where we've had some success in the lab and research-wise, and so now we're sort of beginning to think about therapies and how we launch therapies. So the second part of this is really going to focus more on what we're calling clinical trial readiness. Are we ready to launch a clinical trial if we have a great therapy? So this is going to be less on the science and more on the other things that we need to know in order to get ready for trials. So that's why we're talking about clinical trial readiness. And I don't have so much a talk to give as to begin to engage our panelists, Samantha Parker, Heather Lau, and Florian Eichler, you guys want to introduce yourselves and like say where you're from? That would probably be helpful.
I'm Parker. I'm also from the other side of the Atlantic, as you can tell from my accent, from Paris. Oh, is it not turned on? I didn't. Okay. So I work for a biotech company, a small biotech company called Lysogene, based in Paris, founded by a lady called Karen Ayash, who has a daughter with mucopolysaccharidosis 3A, which is another lysosomal storage disease. And our company is currently in preclinical and working towards the clinic. We're in the clinic for MPS 3A, and we're not yet in the clinic for GM1, but these are our 2 programs. And we are working with Miguel and with Doug on an AAV. So an AAV, one of the AAVs that we use is the AAV Rhesus 10 for GM1, and so gene therapy for GM1.
Sorry. I'm Heather Lau. I'm director of the lysosomal storage program at NYU. I'm a child neurologist by training, and currently I spend about half my time doing clinical trials. I run phase 2 and phase 3, and we're embarking on phase 1 trials in gene therapy eventually. So I take care of a variety of lysosomal storage in both children and adults. I have a very large Gaucher population, so a lot of experience with miglustat and elaglustat trials. And also a large MPS population, late onset Tay-Sachs, Canavan, Pompe. And so that's my experience. My mentor was Dr. Ed Kolodny, who I'm sure many of you know.
I'm Florian Eichler. I'm a neurologist at Mass General in Boston. And I'm a translational neuroscientist. It means I try to move things from bench to bedside. And we just completed a first gene therapy trial in adrenoleukodystrophy using a similar approach with ex vivo gene therapy, as Joe mentioned before. And we're preparing for AAV gene therapy trials for late onset forms, and working also with Miguel and Doug and Cindy and thinking about GM2. And I think, a critical part is understanding clinical trial readiness and what's meaningful to patients. And we've done a lot in terms of organizing the field around patient-reported outcome over the past few years.
Okay, so we're going to, basically I'm just going to put up as a PowerPoint a series of questions that potentially any company or group of individuals would sort of go through to see whether or not we're ready to actually bring something into the clinic. And then I'm going to call on our panelists to answer specific things about that because they have specific expertise. So we'll divide clinical trial readiness, and we're going to use rainbow colors to things that we know. So the reds and the oranges are things that we're more sure about. Things that we're learning, like things that are in progress, are sort of the greens, and then the sort of blues and purples are things that we really don't understand all that well yet and need to spend more time on. So that's the general schema here. So the questions here are the following. How well understood is or are? Now, I put in red the burden of disease. There's absolutely no one in this room that doesn't understand the burden of the disease. I think we understand that very well. So I put that in red. The natural history of the disease and the different subtypes of disease for all our collection of diseases is something that both Florian and I have been working on for 5, 10 years, and we're getting there. So I was going to ask Florian to sort of talk a little bit about different kinds of natural history studies, retrospective, those ones that we look back in medical records and try to pick out, and prospective studies that we use going forward, because these are probably some of the most important pieces of clinical trial readiness.
Sure. So in brief, maybe something all of you know, that in general, the earlier the onset of the disease, the more severe and rapid the progression. And we understand that, but there's a lot beyond that that we do not understand well. And a few years ago, I think through the help of NTSAD and a lot of the families' participation, we were able to dig a little deeper and make use of all the knowledge that parents and families had in terms of recording, their child's life and understanding and remembrance of milestones, and those are extremely critical things when you're developing a clinical trial. Now, why is that? Well, it turns out you want to understand if a drug is making a difference. And in order to know whether a treatment is making a difference, you have to understand what the natural course of the disease is, what milestones were reached at what time, and when were certain functions lost, when did certain symptoms occur. And for the infantile form of the disease, we learned that this was really a very systematic process, and that only about half the children were able to sit up on their own, and but those that did acquired milestones at appropriate times. And I think this is helping us understand where is the window that we can define where a treatment could really make a difference, and where we could bring back function or retain function that allows for meaningful interaction and quality of life. So I'll leave it maybe at that for now.
And actually, it also means that we have to understand what the different subtypes of disease are. You know, we talk about infantile, juvenile, adult onset, and for each of those subtypes, we have to understand the natural history. And, you know, in the early days of NTSAD, and I'm talking about the 50s, when the organization first founded, there was actually some pretty good clinical work, at least describing infantile Tay-Sachs, because that's really what was, and Canavans, because that was really what was being looked at. But, you know, after that, people made a diagnosis, and the physician said, well, there's really nothing we can do for you. You know, take your child home, make them comfortable. So we didn't understand how the disease progressed over time. And if you don't know how things fall apart, you don't know what to call success. And Florian actually put together, how long ago, I can't remember now, several years ago, probably the definitive study on how infantile disease for GM2, and actually it translates pretty easily into GM1 and Canavan, how those milestones really are lost, so that we know that, gee, if you don't lose them at that time with a particular therapy, you say, you know, the therapy is helping. Obviously, with infantile Tay-Sachs, for example, if the children sat up, crawled, cruised the furniture, and started walking, I mean, that would be obviously a positive outcome. But even if you're using a treatment that prevents the children from getting worse, we have to know sort of the pattern of what getting worse is. So, you know, sort of Florian's work with NTSAD has really been important in that regard. Okay, I put in green available biomarkers and their relevance to clinical disease. And we're not going to talk about that so much, except to say that biomarkers often are things in spinal fluid, in blood, in urine, in a patient that you might see as a marker and say, okay, we can follow this over time, and if it gets, let's say, it's a particular compound that appears in the urine, and if that particular compound that is unique to the disease, there's less of that compound with the treatment, sometimes you can make the leap that if there's less of the offending compound, that means that the patient is getting better. And one can use those biomarkers as outcome measures for a clinical trial, but they can't be the primary outcome measure. The FDA is pretty clear on the fact that this has to be clinically, the outcomes have to be clinically relevant. They have to make a difference in the patient. It's not enough to just say, yeah, well, the chemical looks better. You actually have to demonstrate that your treatment is helping patients, like in their daily lives, for example. So that's all we'll say about biomarkers. The other thing before you launch a trial, even in, especially in rare diseases, is do you have a large enough population even to test? If you're looking for big differences, you can do with a small population. But if you're looking at small differences, you actually need a lot of patients. And for our diseases, you know, sometimes there aren't a lot of patients. So Sam has really been doing a lot of work and just trying across the world, really, to get an idea of how we look at what we call epidemiology or incidence of disease. So Sam, do you want to talk something about that?
I wonder what colour it is. Is it blue or purple? Shall I put some clarity?
You're dark blue.
So I think I'm sitting here because I contacted Sue and a few people and panic, you know, what's the incidence, what's the prevalence of GM1? And that's why I'm sitting here, because then I started looking around and trying to get some more information. Why did I want to know that? And I think as we talk about clinical trial readiness, I wanted to know that because as a company, we were applying for an orphan drug designation and a rare pediatric designation. So those are really important. Those are important first steps of proving to the FDA and the EMA. You would use the expression DAR. We know GM1 is a rare disease, so normally it shouldn't be difficult, but we still have to provide evidence of what we're saying is true. So that's the first reason that we want to do that. From a clinical trial readiness perspective as well, what we really need to know is if we're going to set up clinical trials, where do we need to do it, and are we going to be able to recruit patients into the clinical trials? And that epidemiology, that prevalence, that incidence data is also going to give us additional information, as Cindy pointed out, on different subtypes. So we're going to start getting information about how we're going to be able to set up and design that clinical trial, and also where we're going to be able to recruit patients, and also be able to have some kind of clinical trial readiness. So immediately as we move into that stage, we can rapidly, and that's something that's really important for us, because sometimes in rare diseases it takes months or even years to recruit sufficient patients into a clinical trial to be able to show what your therapy is doing. I had another thing also pointing out, coming slightly off the course, but I think it's still really important on the reasons of wanting incidence and prevalence data, is that you want to know, if I know that this is the incidence, well, am I in reality picking up? Are we diagnosing the patients, or are they actually am I not recruiting them, not because they're not finding out about it, but I'm not recruiting them because they're not being diagnosed, or they're not being diagnosed early enough. And that again is a really important role, I think, for these groups, and the work that NTSAD does, is because if we do identify that in certain regions or globally we may not be diagnosing patients enough, then we think about, well, how many resources do I want to invest into improving awareness, so that we can improve on those things. And those are the kind of things that we do, even as a company, on awareness campaigns, in order to be able to help, and that's the data that we get when we know we can collect incidence and prevalence data. So what do we know? So as I was looking around on GM1, I contacted a lot of countries, a lot of people, a lot of registries, and I looked at a lot of publications. And basically, at the end of the day, there was a lot of studies, very, very few studies specifically on GM1. Most of them were on broader areas of lysosomal storage diseases, and they were making assumptions about prevalence and incidence. But we were getting to an estimation that in a country, so I believe in the US, there's 2 million births in the US, something like that? 4? Really? My goodness, my calculations were wrong. I thought there were 2. Well, maybe it's 4, yeah. So you would take a country, France, for example, is a country that has 800,000 children that are born every year, and say we are diagnosing this number of children, let's say 8, it makes it easy, per year. 1 in 100,000 children would then be having a GM1 disease. I've just suddenly lost my track. Let me think. Yes, there we go. We'll get back again. So it's the trouble of not having slides. So in GM1, what we know is in different regions, some people say there's a very, very small population. That's probably because the studies were done a long, long time ago. In other countries, we're having more recent data. The literature basically says there's probably about 1 in 200,000 in GM1. Well, there's a country that has a registry. So in some countries in Europe, we have a mandate, a mandatory requirement now to set up rare disease registries for all rare diseases. So some countries are starting to do this. And a country like France, for example, is able to go back over 12 years and say, go back to the whole of the registry and look at the number of GM1 or any rare disease for that matter that have been diagnosed. And they were able to do that for GM1. They were able to go back over 12 years and were able to provide data. In France, the incidence of this disease is 0.8 for 100,000. So So then you can go back to some of the questions. I'm kind of going to drawing to a halt there, but I think the questions now are in the disease areas that we're working in. So I'm thinking about GM1, but we're also thinking about GM2. Do we know enough about the incidence and prevalence? Probably not. There are countries where we're seeing that, so we should be seeing if that prevalence or that incidence is the same in the US. Are we diagnosing all the children? Maybe. Do we know where they are? No. So there's a lot of work that we need to be able to do so that when we do move into clinical trials, which is going to happen, it's happening, we're ready and families are ready, they learn about it, we're diagnosing them early enough for the treatment, and I think there's, yes, we're blue, aren't we? We need to still do a little bit of work on that.
Okay. Two definitions, because you didn't quite define them, but I think incidence is the number of patients with any particular disease born per so many births in the country. I think you talked about that. Prevalence is how many patients there are out there. So the prevalence, if this was a condition where people lived pretty long, the prevalence would be much higher. If this was a disease like infantile Pompe in the early days, where everyone died by age 2, the prevalence would be only slightly higher than the incidence, how many babies were born per year. So that, and we're talking about trial readiness, we're talking about incidence and prevalence, like how many are actually alive at this point that we can find. And I think you brought up a really good point about how are we diagnosing. And I think the answer across the board, and families could probably echo this in spades, is not soon enough. So, that's going to be another push, that we really have to pick up the families early in order to have the best chance for therapy. Okay. Animal models. I put in red. Are there any animal models? Yes, we know about them. Doug and Miguel have told us about mice, cats, sheep, and we just died. That's okay. I think the next one was also in the red that we knew something about. The course, how well do they reflect the human disease? I think they reflect the human disease pretty well. How much disease correction is needed in the animal models? Well, we know how much vector was put in, or we know how much enzyme is put in, and we know how long the animals survive, so we're pretty okay on that. How much disease correction is needed in humans? We've kind of had that discussion in the last session. Do we want to overcorrect, undercorrect, and how do we hit the sweet spot? So I think that's why I put that in orange. How well understood is the biology or pathogenesis of the disease? I think we know a lot, actually, and it's probably been over the last 10 years that we've learned a lot of different systems that are involved in some of these enzyme deficiencies. But as Sandra was telling us, we need to learn a lot more. And it seems like the more questions we ask and the further we dig, the more huge things are opened up. And in the old days, we used to say lysosomes were the cell's recycling center. The more we hear from the basic scientists at the conferences that we go to is that the lysosome is basically the center of the universe of the cell. So we're a lot smarter than we used to be about lysosomes. Okay, have there been any clinical trials for the disease, and what are the outcomes? I put that in red because we know that. There have been very few clinical trials. Dr. Kolodny had a trial in late onset Tay-Sachs with miglustat. There's been a little trial in pyrimethamine. But, there haven't been a lot of clinical trials, so we know the answer to that question. Are there any other treatments available for the disease, and how effective are they? We know the answer to that, too. Are there any existing resources from which to draw for a clinical trial? And at this point, I was going to ask Heather to talk about clinical trial networks and registries, because the Canavan community has really, begun to address, how to get these things together.
Sure. So I work with a variety registries, both pharma-driven and also patient foundation-driven, and there is an effort with a subgrant from NTSAD to start a Canavan registry, and the importance of a registry. So even if we understand from years ago the natural history of the disease from prior reports, we still need a living, ongoing registry, and there's a couple of different reasons why we need this. In this day and age, we have fantastic supportive care for our patients. Our patients are living longer regardless. And so maybe we'll see a slightly different progression of disease or other symptoms coming out now that we have better pulmonary care, better feeding, nutrition. We're not sending our patients off home to just palliative care. Our children are living quite long lives. And so we really need to chart this progressively, prospectively, how the child is, yes, attaining or not attaining or regressing in their milestones, but what does it look like to live till age 10, 13, or even longer in these classically described infantile diseases? And also to better understand the genotype-phenotype relationship. In Canavan disease, there are reports of milder forms, and there's more severe forms. Dr. Kalani's lab elucidated many of those mutations. So our efforts are to not only to chart the course, but to identify, so incidence and prevalence. So there is an intersection with developing the registries. We're not mandated in the US to do this, and I think we should, and the FDA is encouraging it. Several years ago, I sat down with Dr. Leone at a meeting with the FDA, and that's the first thing they said, was to get together and develop a disease registry for Canavan. And so that's why working together, sharing our information with Dr. Eichler, Dr. Bly in Germany to create an international registry for Canavan is important. So that if and when we're ready to go to a clinical trial for either gene therapy for a disease, we understand the progression. And as Dr. Eichler said, we know when to intervene, and if we're intervening, do we have a signal that the drug is working or the therapy is working, or is that just the natural progression? Biomarkers are limited. NAA in the urine, yes, it's excreted at high rates, but at some point it starts to decline, and that's not necessarily because the disease is getting better. The NAA in the brain that we can measure with MR spectroscopy at some point is no longer being expressed at high levels, not because the disease is improving, but because the brain is atrophying. So it's really important to understand these peaks, those of biomarkers, clinical milestones, and maybe other systemic markers. And so it's important to do this retrospectively. So with Dr. Leone and Dr. Bly and Dr. Eichler, we are encouraging medical records and then going forward. So it's also important, I think we're intersecting with here we are, we need to move this forward, and they're banding together and identifying, I think through another wonderful patient-driven registry, it's more of identifying where the patients are. So if and when we're ready to go, a company that could come in and say, okay, we need to build a clinical trial site here or there because this is where the parent, where there's a huge complement of patients. So it's really important that we work together, we share and that we learn, because these patients untreated can serve as a control population. We have small sample sizes, right? So we can't really say, so I'm doing a late onset myopathy trial for Gini myopathy. It's another inborn error. The difficulty is that we have a finite number of patients and we need to show decline versus stability. And once we start treating the patients, we lose our control population. Now they have been exposed to therapy. So the FDA is becoming more amenable to these historical populations and these placebo or naive patients. We need to do that again now that our children are having wonderful supportive care, to understand the natural history, life expectancies. So again, we're making our efforts in Canavan, but the same goes for late onset Tay-Sachs, goes on for GM1, and any rare disease, MPS, Gaucher actually. And it should go beyond the trial. So I actually hold the registries post-marketing approval. So in Gaucher disease, there's several therapies. The companies are committed to post-marketing commitments to continue to report on safety and efficacy. And as a physician on these advisory boards, we want to see, how does the disease change now that you have the therapy? The example for TIF, infantile Pompe has changed. We have changed the natural history of that disease with our therapy, but now we have a new burden of disease that we have to address. And that's important to understand. Once we've treated you and we now have survival, what other medical problems are going to come to light now that you're living longer? Learning disabilities, muscle problems in the Pompe. And the same thing I can envision with our infantile treated patients. How is their disease going to look on treatment? Because I don't think the expectation is that we're going to basically revert to a wild type or normal. We might have to maybe stop the progression, and then see how else the disease will manifest later in life. So
how to fix and then how to
and
fix
how
those
to fix
pieces.
that.
Yeah.
So I hold these registries, and the data from it is so important. So for Naglazyme, BioMarin is able to report on 10 years of outcome after the fact. Initially, they had a small n, and now we have 10 years of data. The same thing is going to happen with Vimizim. The same thing is going to happen with some other approved therapies.
So I'm just going to make a little shout out for what you just mentioned, which is these diseases are rare. And if you as patients are participating in any studies, I would encourage you to look at the consent form that you're signing and ask, can data be shared beyond this study and beyond this institution that I'm participating in the study in? And sometimes that's just a leftover oversight that many sponsors and companies are not yet incorporating that broad language. But for rare diseases, it's tantamount that your data should be shareable, right? Because that we want to avoid the need for future placebo-controlled trials. We can only do that if we have concurrent natural history or knowledge. Even trials that fail are incredibly important because you really get a definition of disease progression here.
And if you want to go back and use the data after the fact, after the consent form has been signed, actually the institutional review boards or the IRBs at your institution will say, no, you can't share it until you go back to the patient and reconsent them. Right. Well, 5 years after the fact, 10 years after the fact, trying to find the patients, you've moved, you've changed your phone number. And if we can't find you to reconsent you, which is a really big pain, we can't use the data. We can't share the data. So it's extremely important to be able to, and when we say share the data, almost always sharing the data means in a de-identified way. In other words, they're not going to be able to trace you as a person. They're not going to know your name. They're not going to know your birth date. They may know your sex and your age at which you were in the trial, but these are de-identified data sharing, which is becoming quite popular among people in the rare disease community because the diseases are rare. I have a last series of questions. Is there a clear regulatory path? And that would be the path between you're coming up with a therapy in an animal model, being able to take it into a clinical trial, and then being able to, if your trial was successful, move it into a later stage trial, and then eventually to approval so that everyone gets to benefit from that. So have similar drug treatments been approved in the past? For our diseases, not so much. Is repurposing an already is this a repurposing of an already approved drug? In some cases, like the pyrimethamine trial, it was true, or even in the miglustat trial, because miglustat had already been approved for treatment of Gaucher disease. So in some cases, these are small molecules or drugs that have been FDA approved for use for a different disease. It's a little easier when that's the case because the safety piece of this will be well understood, because it will have been looked at in a different population. So there you're more concerned with, is it effective in your disease, not whether or not it's safe, because the people that did the study initially for the first indication have already done the safety piece. Could there be a rare disease designation? Sam discussed that a little bit. There are really big incentives to developing drugs for children, drugs for rare disease, and these can turn out to be quite lucrative if you get an orphan drug designation or a pediatric designation for a rare disease. Companies can actually sell these things for their next trials for hundreds of millions of dollars. So being able to get these kind of designations really is kind of in a way cash in the bank for you to be able to run your trial. So they're very important. And in our case, yes, these are very rare diseases, so this would not be a problem getting an orphan designation for rare disease. What is the estimated cost for the path forward in terms of producing? We talked about producing the vector and how expensive it was, the toxicology studies to see whether or not it was safe, and those have to be done on lots and lots and lots of rodents. The investigational new drug costs, what does it cost just to file something with the FDA? It's not cheap, I can tell you that. I'm learning this more and more. What are the costs of a clinical trial? Part of that depends on what kind of outcome measures you're going to use. Are you going to be doing multiple MRI scans? Are you going to be sedating the children multiple times? You know, do you need fancy, expensive studies that hospitals charge big bucks for? Or, you know, is it going to be less expensive? And clinical trial costs can be substantial.
You want a number?
Substantial.
Yes.
Millions.
Yeah, oh, millions.
Millions.
Oh, millions, yeah.
I mean, all of these are kind of in the million plus or minus range. Safety and monitoring costs. In terms of registries, we talked about this. Many, the FDA in some cases will require that you do follow these kids, a post-marketing registry to see whether or not there are problems. And all of these things, registries, the Genzyme registries that were initially set up for Gaucher and Fabry cost millions of dollars a year to maintain, even after they were set up. So, I mean, none of this is cheap. And I think we're going to have a speaker in a few minutes that sort of speaks to some of the regulatory issues, because it's sort of like trying to navigate a minefield for investigators, for clinicians, and there are people who sort of specialize in helping us get through the process. So I'm going to, for the next 15 minutes or so, open this up to questions from families. So if you have a question, one of us will bring you a microphone.
Thank you for your presentations. I want to talk about what you didn't talk about, which is maybe what you don't deal with, the issue of prevention, the issue of genetic testing. None of what you're doing for these rare diseases would need to be done if there were quality genetic testing. What is your relationship to it?
You don't have a geneticist on the panel.
I can speak to what newborn screening has done for a related disease, adrenoleukodystrophy, in the past 2-3 years. Last summer, it was added to the recommended uniform screening panel. Now there's been screening of all newborns in New York State, as of last year, of California and Connecticut as well. And it's really been a big part of the progress, because with every newborn that's diagnosed, you also are finding relatives that have more late onset forms of the disease. And it's really enriching our sense of the natural history of the disease, but also bringing patients into trials which would otherwise not have been possible. So I think it's absolutely tantamount. As newborn screening only comes about if you have at least some form of treatment available for these patients ahead of time. So you have to show benefit to the screening process. But I'll let you talk about.
For this, it's really a chicken egg, I think, is that if you have a therapy, it's very easy to get newborn screening. If you don't have a therapy, the argument is, well, you're going to discover something and diagnose something in a newborn that that is oftentimes lethal, and you have no treatment for it. Isn't that cruel? That we can't justify doing that. Initially, newborn screening, there are rules set up about newborn screening that say it has to be cost effective to screen the population, and that treating these children is going to provide kids who are healthy and don't have an increased burden on the healthcare system. There are a number of different things that go into newborn screening. But I guess I would also say that our disease, meaning GM2, Tay-Sachs, infantile Tay-Sachs, is the classic example of what's taught in medical schools of what carrier screening can do for the incidence of a disease in a population. I mean, we teach this to our medical students all the time. And this is that in the early 70s, when we figured out what the enzyme deficiency was, Dr. Kabak and others, but he was sort of the prime mover, set up carrier screening so that in the Ashkenazi Jewish population, where this disease was most prevalent, couples were screened before they had their children to determine whether or not they were at risk to have a child with infantile Tay-Sachs. And the incidence of infantile Tay-Sachs in the Ashkenazi community within 10 years dropped by 90%. It is the classic example of what carrier screening can do. Now, the difficulty has been into trying to generalize that to say, well, okay, now we've got the Ashkenazi population pretty much taken care of. There are some caveats, and you guys have probably heard me talk about this before. given the fact that this is now the United States and there is intermarriage between the Jewish population and non-Jewish population, sometimes your genes are Ashkenazi Jewish, but you don't identify that way and you don't identify with that community and you're not in places where there's carrier screening. And so, oops, you didn't know and you didn't know to ask. And so now you are sort of joining the rest of the population with your carrier risk. And there is a baseline risk of Tay-Sachs, GM1, Canavan that really speaks to the need for doing generalized carrier screening sort of across the population. And that's not been mandated in our healthcare system yet. So it is possible to get that screening, but families usually have to pay for it. So there are moves, and there have been moves within this organization to advocate for more generalized screening of everybody. So I think we're talking newborn screening, we're talking carrier screening. It's not exactly trial readiness, but I think it does speak to incidence prevalence of the condition in the population. Any other questions? Yep.
So speaking as a family member who has raised money from friends and family to fund research through NTSAD, I'm struck listening to what I consider just an extraordinary series of speakers talk about the whole spectrum, which I really appreciate, that there are so many competing needs for so many limited dollars. You know, like research, clinical trial readiness, management of the patient registries afterwards. And it is a little overwhelming. And I'm just trying to think about how as a family member or anybody who is raising money, do we think about prioritizing? Are there like 3 priorities all at the same time? And I wonder how to think about this.
So maybe you know.
Yikes.
So when we're working in industry, we have our development. We have what we call our critical path. And I think that maybe in this area we need to think about what's our critical path, what's our aim. Our aim is to get to families as quickly and safely as possible. That's our aim. So what's our critical path to do that? And along that, we're human beings. Human beings always have, that's interesting, and spin-offs and things. And they're good to do because you learn, but you just have to, when we think we're sitting around a table, and despite we did an IPO, we've got some money, we still have to think, well, it's all dollars. So we say, somebody will come up with a bright idea and go, yeah, that's really great. And then you say, hang on, it's not on our critical path. It's not on our critical path. We have to refocus. And I think that's the way of defining within this group what's NTSAD critical path for this goal, which is getting our treatment and focusing on that.
I'm going to comment. As an investigator that works with pharma but not pharma, it's awfully frustrating for me to see the diseases that have a potential drug, how much effort goes in from the companies. Once you get a company interested, then all of a sudden it just opens up these wonderful avenues. Like the Genzyme registry is such a robust registry that this company has decided to put millions of dollars in, and it's amazing for Gaucher disease, and we're still learning about Gaucher type 3, type 2. And then I look at Canavan and I think, I was introduced to this by Dr. Kalani several years ago, and it's really frustrating that if there's not a drug on the horizon, there's no attention towards it from a pharma community. And so the problem is that when pharma gets interested, that's when they're starting their natural history study. And my point of view is, could we start this sooner? But it takes money, and that's where the NTSAD seed grants are so important. Yes, I'm advocating for this aspect of it, but these little seed grants are so important to just kind of push a couple of investigators together, Dr. Eichler, Dr. Bly, and myself, and give us a little support that we can put a lot of effort into that until we can push it even further to get interest from pharma.
And that would be exactly what's on our critical path, would be the natural history.
They need that.
Because,
they need
as you said before, when we talk to the regulatory authorities now, we are able to do without having a control group. So that's our aim. That's our aim. We want to treat families. So not having to go into a clinical trial where you have to think about having a group of kids that are not receiving treatment, which is, from every perspective, you need that natural history. So that's critical path for us.
I'd almost say NIH should support us to do this, so that we are ready for when it's time.
So I just want to pick up on that one word you said, sort of competing, and I think actually what you're looking for is synergistic projects and trying to understand the entire landscape and look for alignment. Because if you can do that, you can save costs, and you might be able to hit the ground running with certain projects if there's already something that's happened in a related disease. I mean, we learned a lot from the ALS field. We really didn't have to build our first repository ourselves, because we borrowed and stole their diseases. And I think that's really critical to understand where are enough synergies that you can make use of them.
And in terms of borrowing and stealing, if you can borrow and steal from diseases that are more common and more visible in the population, i.e. Parkinson's, Alzheimer's, it's a way to leverage. Kevin?
Just kind of building off that question, which was a really great one. So we talk about the 20-year journey that could possibly happen, as Miguel mentioned, as we start exploring all these things. But with the aim and with the critical path mentality, if the FDA came to us tomorrow and said, okay, you can do clinical trials right now, use your registry. We need you guys to be able to tell us how ready the registry is and where do we need to do more work on that registry that is critical, so that we don't make any more delays in this process when the opportunity comes along. So whether you can articulate that now or when you go to your SAC meeting later, we ask you guys to really spell this thing out, because I think we're on to something very, very special here, and we just got to dive all over this. So thank you for focusing on that.
It sparked something when you said that, because I mean, what I would reply to that is what we need. It's not just data; it's the quality of the data and the integrity of the data. Yes. Okay. So, for example, when we're doing our natural history study in mucopolysaccharidosis 3A, we're mirroring what we're going to do in our clinical trials. So we already have a pretty good idea what our protocol for our clinical trial is going to look like and what assessment tools we're using. And we're doing the same thing in our natural history study, but before, when we're still finishing all our preclinical and doing our manufacturing, so we're not delaying treatment, we're doing that. So, and then what we're doing for that as well is, that's why they cost a lot of money, is because we're putting all the processes in to make sure that the data is absolutely top quality. We've trained all our neuropsychologists, we've got all our data management people going in, auditing the data. It's not, and so, you have to weigh between wanting to get lots of data or wanting to get a small group. The study that Cynthia's done, I mean, is brilliant. It's excellent. It's a small group, but it's great quality data, and that's the kind of stuff I'd say we need.
And I think from my perspective, that's where we saw an unmet need to organize this data that's out there for Canavan, and to try to collect prospectively would be the next phase, good data. And that means this has to go through a consent process. This can't be crowdsourced on social media. Social media is wonderful for you, but as a physician scientist, we need to gather data in a ethical and regulated way through consenting, and then to make sure that we have good data, actual reports, MRIs, lab results that are from a valid lab, and then look at that over time. So we are just beginning, and it's a lot of bureaucracy, but that's how it has to happen. And thankfully, the entire platform was donated to us through Neurobank. That, I don't even know how much that would have cost. So for us, before pharma gets involved, we're trying to figure out how to organize it, get the patients and families engaged when we're ready. The database has just went live. after a long time. And so that's where we're at. And I would love to talk with the families later that are here for Canavan, and we can give you an update on that. And then the next phase is to add to it is the prospective, where we have patients come in and do systematic exams prospectively. So that would be the next phase for Canavan specifically. You can speak to, if you want.
I've got a question.
Question?
Yeah, since being diagnosed with late onset, I've constantly heard the limitations financially for getting support to do the research and so on and so forth. And in my free time, I spend a lot of time on the internet looking for information and seeing what's out there. And I came across the CRISPR treatment, where Jennifer Duda at University of California, Berkeley, where they go in and they adjust someone's DNA. And I want to get your feedback on that. I know it's significant because it's something that can affect a lot of different diseases. And for that reason, there seems to be a lot more money going into it for treatment. And I just wanted your feedback on that.
Outside my area.
So
Anybody want to tackle it?
I can do it in layman terms because that's my level of knowledge. But I think so. Gene editing technology is likely here to stay and will continue. I think the gene therapy platforms we're pursuing are going to be what gene editing will work on top of. CRISPR-Cas9, they're like, it's like molecular scissors. They right now they're pretty good at cutting out genes that we're maybe not quite there in terms of replacing. So I think it will first work for those diseases where you have so-called gain of function, where there's some toxic effects where you might be silencing, cutting out, and that might help. I do think there's probably some value in having an endogenous promoter, so having your own promoter that's regulating your gene work here. So there might be other fine-tuning aspects that will be the future. But I think what we're doing with gene therapy is going to pave the way for that. So I don't know. You're far smarter people to talk about that.
I think that the whole CRISPR thing is really in its infancy. It has great potential. As far as human applications at this point, I don't think we're there. I just think there are a lot of kinks to work out. But going down the line, I think it's probably where gene therapy was. Tell me, 15 years ago?
20 years.
20 years ago? But hopefully it will move faster than. It will move faster than.
It will move faster.
Because gene therapy hit a significant bump in the road and was derailed for a while.
There are actually 2 clinical trials already using CRISPR.
Mm-hmm.
It's in.
You get 2 seconds. And then
There are 2 clinical trials for CRISPR.
and what are they in?
They're modifying, I think, bone marrow stem cells for cancer therapy. And I think one of them for cancer therapy, and I think one of them is for HIV as well, right? Is that the case, Joe?
Not a trial yet. They
Or
have
it's-
zinc fingers.
Yeah.
So zinc fingers are similar to CRISPR, where they're
Oh, sorry.
So there's a clinical trial for HIV using zinc fingers, which is another gene-modifying enzyme. It works similar to CRISPR. So Sangamo in California is doing that, but not CRISPR for HIV yet, but similar gene modification enzymes. So it's actually moving a lot faster, like Miguel said, so it probably won't take 20 years, because the zinc fingers were started maybe 5 or 8 years ago. Sangamo started it, and it's already in trials in humans.
Yeah. Sangamo is actually moving forward with that in a couple of different areas, and you'll see in the next 6 months some clinical trials first in human.
Okay. I have, actually, since I'm going to take prerogative in the last 3 minutes before we introduce our speaker to give you 3 more slides. As a shout out, when we talk about clinical trial readiness, we actually talk, and really you saw from my title, I think you saw from my title, was how can patients participate? So now I'm going to give you an example of how patients did participate. And it's a lot of work. And some of you who participated are in the room, and you can tell your colleagues and your friends how much time you did spend participating. So this is a trial Actually, this is I hope a success story of something that NTSAD funded 1 year ago. 1 year ago, it was nothing. We had just gotten the money to run not a clinical trial, but a natural history piece using, trying to think, activities of daily living, sort of real-time data from real patients that would make a difference and could possibly be used as outcome measures for clinical trials. So through NTSAD, with funding from the Burek family, we took $20,000 and basically at the NIH were a pass-through to a company called Apparito, who have actigraph watches similar to Fitbits and an app that they put on your phone that monitored events, that allowed patients to complete outcome surveys and medical appointments and I think drugs. So we invited 8 patients with late onset Tay-Sachs and Sandhoff to come to the NIH last August, and we put the watches on. These are the little watches. There are better versions of them now. There were some issues with the watches. People can tell you about these. And basically, we just now, after 6 months, invited everyone back and did some more testing so that we could correlate what we were measuring in the clinic against what the watches were telling us in the field, basically. And we enrolled 8 subjects. They wore their watches for 6 months. All of them had their apps installed on their own personal cell phones, iPhones or Androids, and they had the watches. And they wore them pretty much all the time, except over the holidays there was a little bit of a dip in watch wearing. So this is one of the subjects of the 8, and so you can get an idea for the magnitude of how much time and effort people did. People wore their watches all the time, pretty much. This particular subject reported 83 events, 30, we'd asked them to record choking and near choking events, other illnesses, if they fell, and other issues like headaches and other things and tremor. So this particular patient reported events 83 times into his phone, made 66 visits to healthcare providers and documented which they were. And then we asked families or patients to do what we call PROs, or patient-reported outcome surveys. These are surveys that looked at how their disease impacted the wider family. We asked them to tell us about their tremors. We asked them to tell us about fatigue, about how well they were thinking, about how fatigued they were, how well they slept, how they felt about themselves. And so these patient-reported outcome surveys were pushed to their phones at periodic intervals every 2 or 3 months. And this particular patient, and I would say the vast majority of the patients, completed 82 of these surveys in 6 months. So the takeaway message here is, yes, patients can be involved in clinical research. They are critical for us understanding what are real outcomes. When I talk to the FDA about this kind of outcome measures, you know better than I do, in late onset Tay-Sachs and Sandhoff, the 2 big things are speaking clearly and being able to walk and not fall. Well, if I can record how many times you fall in 6 months, and now I institute a treatment, and you're not falling that many times in 6 months, that's an outcome measure that actually makes a difference. You're not falling on your face in the middle of the street and really hurting yourself. I mean, that would be real success. And that's the kind of thing that the FDA is looking for. So my take-home message here is, and that's really the end of it, my take-home message here is that patients are critical for trying not only to participate in registries and going through clinical evaluations like we were doing at the NIH, but also just on a day-to-day basis telling us how you're doing in a way that we can record the data and use it for clinical trials. So with that, I think we have one more speaker who's going to tell us about regulatory, I forget the first name, Schwartz is your last name?
Cordula.
Okay.
I'll introduce myself. No
At
problem.
any rate, we're going to hear about the process, the regulatory process for getting things actually to happen.
That's right. So my name is Cordula Schwartz. I work in the Regulatory Affairs Department at Sanofi Genzyme. And I want to thank Sue for inviting me to speak about the drug development for rare diseases. So this will be kind of an overview of everything you heard already, and I'm going to put it all together for you. So I'm going to talk about the drug development process, what a standard trial design looks like, how this compares to a trial design in rare diseases, the importance of natural history studies, what kind of expedited programs we have available to us from the FDA, and how that then translates into the drug development process in rare diseases. I have a brief update on the 21st Century Cures Act, and then conclusions. Oh, there's animation. Did you?
These are all PDFs. So
oh, they're PDFs. Okay, there was some animation. That's okay. So this is a very simplified schematic of how we develop a drug and how it gets to approval. It obviously starts with the drug discovery, and where we identify a potential candidate, and that, you know, the time that takes could be very long, and that obviously depends on the disease and the drug. And then we heard this morning, after you identify your candidate, you have to test it in animals. So we heard at length this morning how long these studies can take. So you need the right animal model, and you want to show that the drug actually works, that it has the treatment effect you're looking for, and that the drug is safe in the animals as well. After you've shown that, you need to talk to the FDA, and then you can go into humans. So you start with your clinical studies. They're divided into different phases, and phase 1 is your first study, mostly done in healthy volunteers, where you look firstly at safety and then at the dosage. And we talked about that this morning too. You don't want to have a dose that's too high, and you don't want to have a dose that's too low. And you show, you know, that you see that your drug is doing the desired effect. So you have target engagement in your drug in phase 1. These studies can take several months, and 70% of drugs that go through phase 1 move to the next step. The next step is phase 2, where we look at early efficacy and side effects. And these studies are smaller, and they take several months to up to 2 years. And then if those trials are successful, 30% of those drugs will then move to the next step. And that will be your phase 3 studies, which are your large efficacy studies with hundreds to thousands of patients, and where we look at efficacy, and we'll always monitor for adverse events. And these studies take 1 to 4 years. And then again, 25 to 30% of drugs move to the next step, which would be your regulatory submission and approval. And as we talked before, oftentimes we do have, we continue with studies after approval. So that's a very simplified version or picture of what the drug development process looks like. At the same time, anything that moves into humans, the preclinical work is important for phase 1, but we continue with animal studies while the studies are ongoing, because for submission we do need additional toxicology studies. We need to do certain requirements, and same for manufacturing, which that's not depicted on the slide, but it's a lot more complicated. So I wanted to look a little bit at what a standard trial design looks like. So the FDA is looking for adequate and well-controlled trials. So what does that mean? They look for hard clinical endpoints, and that means we need to have a good understanding of the disease so we can actually measure the desired effect. And we want to see meaningful improvements for patients and their families. And the other very important piece of a good designed trial are the controls. So we want to make sure that the drug's effect is real and not by chance, right? So we do blind investigators and participants to treatment. And we have a control arm, which means some patients get the drug and other patients might get a placebo, which is a non-active treatment, so that we can see whether this effect is really real. And we do conduct the trials at multiple study sites and also in multiple countries. Usually the agencies require more than one phase trial. And obviously you need a large number of participants, and we talked about this earlier too, in order to see the effect and to be able to measure it in a statistically significant fashion. You need to have the right amount of patients. And obviously you want to reproduce your results. So in rare diseases, we talked about this earlier, so we still want trials that are adequate and well controlled. However, as we talked about, endpoints aren't always defined and been, previously recognized with the agency. And that's because some of these diseases are poorly characterized, or there's we have varying rates of disease progression, and the diseases are very heterogeneous. So it's difficult to find an endpoint that would fit everyone. And in order to help that, we could use what we call a surrogate endpoint. That means it's a stand-in for your hard endpoint. That could be a biomarker, which we heard this morning. It could be a chemical that you can measure in the blood, in the urine, or in the spinal fluid that might change with treatment. But the important piece is, does that really then mean that your drug is clinically beneficial? So there is a high bar for us to have a biomarker for what we call an accelerated approval, so that the agency agrees, yes, this is predictive of clinical outcome. But even if we get approval with the surrogate endpoint, we still need to do a follow-up trial where we can show clinical benefit. And you might be familiar with the Sarepta drug that just got approved. They got approved under accelerated approval by showing lowering of dystrophin levels. And they do need to do this follow-up study now. So controlled, we do want to control the trial. And if we show benefit in an early trial, we have a dilemma because we don't want to put patients on a placebo if we think the drug is working. So again, this is something, where it complicates the trial design. So in that case, if we have historical control groups, those natural history studies, we can use those as a control arm, and we don't have to put patients on placebo. And we know, we talked about this, there's much smaller number of participants for rare diseases, so that is another challenge. So natural history studies are very important because this will help us understand the disease pathophysiology. So we heard about this already, so we could maybe identify different disease populations and subtypes and different, and we can measure how patients are progressing. Because when we design a trial, we want to make sure we can measure an effect in a reasonable amount of time. And if patients are too severe, we might not be able to measure change or improvement. If patients are very mild, it may take a long time to show an improvement. So we need to find a sweet spot on which patient to include in a trial. So that goes into, why can some patients not participate in some trials and other patients can? It's very important because we need to have a population that's not too heterogeneous in order to really be able to measure something in the clinic. And as we just talked about, and Dr. Tiff just explained with this wearable device, you can, with these natural history studies, sometimes find endpoints that you can then use in a clinical trial, or specific assessment tools that can be agreed to with the agency that are very disease specific and that are designed to just measure clinical benefit in that different population. So at FDA, we have several pathways available to us to expedite the approval process. These are called expedited programs for serious conditions, and rare diseases usually fall into that category. Fast track is one designation we can achieve if we can show benefit in animals. So it's just based on efficacy shown in an animal. And they have actions to expedite the development and the review process. And you can submit your submission in pieces. It's like what we call a rolling review. So you can submit bits and pieces and they can start reviewing, and that kind of speeds it up. Then breakthrough therapy, you probably heard about. You have to show a benefit that's clinically significant in humans to get this designation. So that would be an early trial, like a phase 2 trial, or a phase 1 potentially. And you can have intensive guidance from the FDA and lots of interactions that are very available to talk to you, to guide you through your development process. And also you can submit your submission in a rolling fashion. Accelerated approval is based on the biomarker or surrogate endpoint that I mentioned earlier. It needs to be followed by a clinical study. And then priority review shortens your review time by 4 months. So oftentimes, if there's no drug available, the FDA will grant you priority review, and they say they will review your drug faster than a regular drug development. So here's what this can look like now if we look at all the other ways that we can accelerate the approval process and the development process for rare diseases. So we still have drug discovery, we still have the preclinical studies. That's the same, right? That hasn't changed. That's the same if you have a regular development or not. Phase 1 is still the same. So you need to first show safety in humans before you go into your next trial. But then we're trying to get creative, and this is a potential scenario that needs to be agreed to with the agency, of course. So you have agency interactions at all these steps when you go from one phase to the next, where we could potentially combine a trial that we don't do 2 steps, but we could do like a phase 2/3, where we can show efficacy and in a randomized and controlled trial. So I think what's really important in the rare disease world is to do controlled trials early and not do an uncontrolled trial that may look very promising and then can't be reproduced in a controlled trial. So if we can combine these and just do controlled trials early, I think that is definitely needed in terms of speeding up the process and getting early results. So again, we can use potential biomarker endpoints that would speed up the approval process. And these trials can last anywhere from 1 to 4 years, depending on the disease. So then, like I talked about, the regulatory review process can be shortened. And then we still have post-approval studies for the most part. Sure.
So simplistically, why would it take 1 year versus 4 years, and what do we need to do now to make sure that we're on the track towards a 1-year solution and not a 4-year situation?
So I'm not sure if I'm qualified to answer this. It's probably better to be answered by the clinicians. I think it depends on what you need to measure, right? On how fast your disease progresses and how and in which time frame you can measure a difference. So for neurological diseases, it's challenging because those endpoints tend to progress slower, and changes are more difficult to measure. So I think that is where you see the difference. I'm like, what's an example? I'm thinking about, a diabetes trial or something where you may have a 6-month endpoint sometimes because you know exactly what you're measuring, you know how fast your glucose levels change. You have that very defined, and in our cases, it's not as defined. So I think that sort of weighs into how long these trials take. And I don't know if anybody wants to add to that.
Yeah, Kevin, it's not so much can we fast track it. It really is whatever your outcome measure is, how long it takes to show a difference. So for example, if you were going to use infantile disease and it progresses very rapidly, you might be able to show a difference in 6 months to a year. But if you had juvenile patients who change more slowly, or late onset patients, it might take longer to show a difference. And you actually set that up when you set the trial up at the beginning. It's
Correct.
not like how much longer do you have to stretch it out. What the end, whether it's 1 year, 2 year, 3 years, 4 years, when you start the trial.
Right, and that's very important. Your endpoints and your measures need to be defined before you start your trial. That is the key. You cannot do a trial. And this is sort of where it's complicated, right? Because you don't know for sure, especially if it's a disease that hasn't been well characterized or you don't have precedent. Go ahead.
It goes back to the natural history informs your endpoints.
Yeah.
So I'm going to harp on that. So if it's a slowly progressive disorder and you want to try to find something that changes within 1 year because pharma doesn't, or 2 years because it's very expensive. So the same thing, again, my experience with the genie myopathy population, phase 2, is what can you see in 1 year? But in other disorders, we didn't show a change, so we had to go to phase 3 and do another placebo-controlled trial, even though we saw a little uptick in stability. So your natural history informs your endpoints and tells you how long you need to do the trial. Certain trials might be aborted before you could see it, and that's unfortunate. Cognition or changes in memory or changes in learning might take a little bit longer time, and you wouldn't want to stop the trial before you gave enough time. So,
Correct.
It has to be predefined.
Yes, it has to be predefined, and endpoints need to be agreed to with the agency, right? So this is where, I'm the link between the FDA and the team at the company. So you need to agree to these endpoints before, and you need to kind of estimate how long does it take for them to change.
So the reason, and I mean, everything you said makes sense, but what we as families think about is, if there's a clinical trial and there's a finite group in the clinical trial and somebody's not in the clinical trial and it's a 4-year duration, if we can treat people in the 4 years that need to be treated, and we're learning and we're verifying that all the endpoints are being met, that's one thing. If we can only do a few people and we wait 4 years for the next group because you can't expand for approval reason, that's a very different concern. And I think we need to take a practical approach to that.
Yeah. Yeah, you can.
In 1992, the first Genzyme drug for Gaucher required only 12 patients and 6 months follow-up. 25 years later, and we know what happened with Fabry, we know what happened with other diseases. Okay, so it's not really change. It's a change in environmental requirements, partially, I think.
Yeah.
No,
I think, I'm thinking about, okay, we had the Sera-Esteves at the time, the Sera-Esteves trial, and then we had the eliglustat trial, and the order of magnitude in terms of patient numbers is enormous.
Yeah.
So I think.
So I have kind of 2 questions. One of them, after the Century 21 Act, do you see any reversal of that trend? And then follow up, Kevin question, how do you address it with patients? For example, late onset Tay-Sachs patients, how many patients would you need to prove initial efficacy or safety? Okay, it's probably more of a safety trial. And Cynthia, I see you kind of probably wanting to answer my question.
It depends on, the number of patients that you treat. Is that it depends on it depends on your outcome measure. And it depends on how many patients you start in your trial. Part of that is dollar driven, and part of that is how many patients do you absolutely have to be able to show a significant difference. If you only have 2 patients, but they do hellaciously well, okay, fine. But if you're looking for a small change, you may have to do 20 or 25 or 30 patients in order to statistically demonstrate that you've got a change.
I can go into the 21st Century Cures Act if you want me to. That might answer your question. So as you guys know, the President Obama signed into law the act in December of 2016. And the purpose of the act is to modernize clinical trials and incorporate the patient perspective, because they realized that this is definitely needed. And I pulled out 3 of the subtitles that I felt were most relevant for the development in rare diseases. The first one is patient-focused drug development, and FDA had a mandate to collect patient perspective, and that actually has been ongoing since 2013, and it ended in 2016. They had 20 meetings, the voice of the patient, one of which was neurological inborn errors of metabolism, and some of you may have attended. And so they want to look at new approaches on how they can collect the patient experience in a clinical trial to help, come up with an endpoint and a meaningful change for the patients. And so they are required to put together new guidance over the next 5 years. And then incentive programs to advance new therapies, so one of which was mentioned earlier, it's the Rare Pediatric Disease Voucher. So this is an incentive program for companies or anybody to develop drugs in pediatric patients. And they expanded the orphan drug grant to fund observational studies. And I think earlier we mentioned also this is not part of the 21st Century Cures Act, but there is an incentive already in place that was from the Orphan Drug Act that was put into place, I think, in '92. It's been around for a while. So if you can get orphan drug status for a disease that's very rare, it's less than 200,000 patients in the US, you have some tax incentives and filing fees reductions. So there are incentives out there for companies to develop those drugs. And then they are also looking at modern trial design. And so they want to look at novel clinical trial design. It's kind of a vague term, but one of them is, one of the things that were mentioned earlier, if you have a drug that's already approved and you repurpose it for another disease, how can we include the real world data into a new approval, right? How can that be integrated without having necessarily to do like a whole nother big trial? So I think that is where this is going, and they also have to issue new guidance over the next 5 years. So I think, to answer your question, I think they're trying to work with us. I think, it's, and they're very, they definitely are trying to, they're listening, I think, and they know that the patient perspective is very important. So just to conclude, we know we have our challenges in rare diseases, and natural history is invaluable to designing good clinical trials. And we definitely can make use of incentive programs, and we have expedited approval processes available to us. And like I just said, patient-centered drug development is so important, and your voices are very important. And we do believe that health authorities do listen, and we're working towards speeding things up. Thank you.
So we have 5 minutes. I don't know if there were any other final burning questions that people wanted to ask. Okay, we have.
Sorry,
this might be for you, not me.
As a parent, my son passed away last year of GM1 infantile form. This is kind of a question, I think, for everybody that spoke today. Miguel talked a little bit about it, but it deals with the clinical trial for GM1. I know we talked a little bit about timeline. Now I realize, by listening to you about how all of the necessary steps, I just didn't know if we could get more specific on kind of where we stand with that. If anybody could talk to us a little bit as parents, where we stood with the clinical trial. Doug, Miguel, maybe.
Is there anything?
Hi, Sarah.
Hey, Doug.
Our timeline that we set forward for the IV gene therapy trial for GM1 is pretty much on track, and end of this year, early next year is what we're targeting. What we always say is that making this vector is a biological process, and there can be, biology does what it wants to do sometimes, so there are no guarantees, but nothing that we have seen or been told to this point has led us to believe that we can't make that timeline. So end of the year, probably January 2018 is reasonable. Yeah.
So this is really great news, because potentially there's 2 clinical trials in GM1, right? So I'm working with Doug and Miguel, who's also working with Lysogene on another approach. I'm always very reluctant. I mean, maybe we can talk out of the big room as such on a more personal level. It's always difficult to give timelines. I mean, I've got a timeline in my head, and I know what our critical path looks like. It's also balancing between moving into early stage clinical trials very, very quickly, and then having time spent redesigning things, sorting your vector out, your manufacturing and things like that, and having that big time before you're able to move into your more final clinical trials, which actually, at the end of the day, might be more time consuming than actually getting everything sorted, designing a trial where you're not looking at your stage trial, but you're looking at more of an adaptive design, and you're able to move in and change things around, change patient numbers and things around, and getting all of that really right. So it might be a few months later, but you might actually, at the end of the day, be much quicker getting to, because clinical trials, as we're discussing, it's a limited number of kids. We're talking about early access programs as well, so that other families don't have to wait. The small number get into clinical trials, and there's all those other people that want therapy, and it might be too late by the time we get to marketing authorization FDS. We need to think about all of that. We need our manufacturing sorted in order to be able to do that. And then we also need to design it so that we can get to market authorization quickly. So I haven't given you a direct answer because I prefer not to in this kind of stadium as such. But you know, it's a very good question, and we do have a clear pathway.
Okay, thank you. That was a, we have, Emily, can you take the mic back there so we can? No.
We have to record it. We have to have it on.
Wait.
Okay. Thank you. I've been a teacher for 30 years, so I've got a big voice. But I have a similar question, and I understand your answer about not being able to be specific about a timeline. But my question is about, and I'm going to ask, timeline for clinical trials for late onset, and where Genzyme or where this gene therapy program is. I know you're starting with a different rare disease, but where does late onset fit in, and is there a timeline?
Is there a timeline? Would you like to comment? Or would you?
The person who can... Anna, comment.
Hi. So I'm Anna Pogam, the clinical lead for a few of these programs. And when you say late onset, I'm assuming you talk about Tay-Sachs and Huff, right? So timelines, as you just heard from Cordula, it's more than a wish, right? So we have been working for the past 3 years, and some of you are going to be participating later, on some of the assessments exactly to try to fit into what endpoints are reasonable and things that we can show that change. As Cordula alluded to, it's a very lengthy process, actually, to get that agreement, because even though we have collection of information, we still have to prove that that's something that holds to the standards that we're asked to do so. So even if, yes, we can look around and see, yes, if you can improve walking, or if you could improve tremor, or if you could speak a little better, makes sense. We also know that there has been failures in the past. So for us to be able to provide a timeline, which we have been working very hard for the past 2.5 years, we have to get those pieces of agreement with not only FDA, because let's also remember this is going to be of global importance. So in our timeline, as much as we can push for that, without having any endpoints that are agreed upon with any of the regulatory agencies, we are actually gearing up, as was mentioned before, for early consultations with them. Not only FDA, but also with the regulatory authorities outside. And that triggers, if we have enough, in the case of this particular product that might be available, that might be accepted to be treated, we're gearing up to having those consultations hopefully before summer, or at least at the very beginning of summer. From then, it's a clock that unfortunately is not in our hands, as pushy as we are, but it's not in our hands. So that goes into what Cordula was alluding to, hopefully trying to get combination of phases instead of all these many steps, because we don't have neither time nor enough patients to do that. We don't have 1,000 late onset patients to go around, right? so that is all a negotiation that actually needs to feed into the properties of the molecule itself and what needs to be presented. I also wanted to link that with the natural history that has been mentioned several times already, but also with the patient-reported outcomes, right? One of the difficulties is that even though you might be very aware of what would be important, you still have to produce something that can be used by anybody, anywhere, and is going to be perceived by the people who actually have to pay for the drug, which tend to be very expensive drugs, that is capturing something of value. And that's the difficulty with natural history, because to start off, you're gathering information. You don't know what's going to work out. So it's always much easier to think in hindsight, I should have asked the question X, Y, and Z. So that's also why we started with a very, very broad net of anything that could potentially help, right? Even if it's not 100% every patient has it. And this is where the individual stories of the patient, until you can get into a repository that's not necessarily linked to any company, most of the registries, and I should say natural history because registry has a lot of other interpretations, but natural history usually starts in one place with a handful of patients, with a physician that follows those patients, right? So all this needs to play into trying to accelerate. What Cordula mentioned as well, maybe a biomarker can be a surrogate, but that's a step. That's something to try to accelerate. You still have to prove that it changes something in a real person who is suffering from that. So even if the timelines are there, and as I said, we're aiming to do that for this particular group of diseases right before the summer break, we still need to go through all that. In our case, we have enough data, we hope, that's going to be able to link that on the mechanism of the drug itself. But there is zero in terms of the specific disease, right? So we need to reach again, start from scratch with each of these regulatory authorities to inform them, this is why it's important, this is what it causes, and this is what the patients would like to have. This is a measurement that it holds. It doesn't matter where you come from. It's still possible. It's a very long-winded answer, but it's just to put in perspective that it's beyond the wish. It's beyond looking around and knowing, well, this is what you should change. You still get the exact same questions because, as Cordula also mentioned, there's a very structured path that needs to be followed. And sometimes, even if the need is there, you still have to demonstrate that, yet again, it works. So that's the best I can provide you, but we're hoping that at least knowing the previous diseases that have been worked with, this particular compound, we're going to get a leg up, I think.
Thank you so much. We do need to wrap up, but I want to thank really all the speakers, our moderators. Everybody's going to be around at lunch today, so the whole point of them being here, other than being up on stage, is that you have a chance to talk to them one-on-one. I am going to pass around some evaluations. It would be great for you to fill them out. One comment, one observation is that I think for me, this is I think my 9th or 10th conference. The conversation we're having here today about research is very different than the conversations that we had certainly 9 years ago and maybe even 2 years ago. And I know it's not as fast as we want it, but it's still, I think a year from now, it's going to be a different conversation. And so I think it's very hopeful. One other thing, thank you to the Matthew Forbes Romer Foundation for making us available to do this, making it possible. And of course, thanks to all of you. I know many of you have raised money for research. Some of our research funds and families are noted in the program, as well as, of course, all the other foundations that are also working hard to advance this. So thank you, and lunch is around the corner.